As your system partner, b.braun offers a unique portfolio which covers the entire process of nutrition therapy, including the required medical devices. Meeting the demands of each patient, we aim at improving life together. Whether in the intensive care unit or in the hospital ward, whether in pediatrics and neonatology, or at home. Together with our customers, we can establish a state-of-the-art nutrition management in each hospital. By offering enteral and parenteral nutrition products, we support physicians with numerous options to create regimens that are adapted to their patients' needs. Benefit from our value-added services and applications, such as discharge management, home parenteral and home enteral nutrition, as well as reliable, up-to-date information on compatibility based on our EasyComp application. The Aesculap Academy is one of the leading medical education forums for everyone who is professionally, passionately, and ambitiously committed to people's health. In close cooperation with the Aesculap Academy, we run workshops and trainings, like today's NutriTalks. We constantly seek constructive dialogue with patients, doctors, and healthcare professionals in order to gain insights for progress for treatments and routines in this field. Our guiding aims: reducing risks, simplifying procedures, enhancing safety. In cooperation with Aesculap Academy, we want to provide with NutriTalks a platform to the newest information on nutrition in ICU, pharmacy, and along the patient pathway. In addition to the lectures, there will be more information available in the virtual exhibition hall. These and further topics will be presented at the NutriTalks event. Welcome to NutriTalks. The b.braun Sharing Expertise means continuously developing effective solutions through constructive dialogues with our customers. This virtual scientific training event will consist of lectures from key opinion leaders, medical scientific affairs, and GMS, complemented with an attractive exhibition area. Let's have a look at our program for the next 2 days. We have clustered the lectures in 5 different sections. The topics covered will be Pharmacy Nutrition Compounding, with 2 contributions regarding compatibility and automated nutrition compounding. Nutrition in ICU, with 2 sessions about the benefits of omega-3 and current guideline recommendations. Home Artificial Nutrition, with 2 lectures concerning a 21st century perspective and the role of vitamin K. Nutritional support in specific clinical conditions, in which 3 contributions will inform you about preventing malnutrition in ostomy and chronic wound patients, intradialytic nutritional strategies, and omega-3 in oncology. And last but not least, the section Protein and its Clinical Relevance, with a lecture regarding clinical practice with Vita Limed. On the second day of our event, we will cover the same topics in reverse order. The recordings in the entire platform are available for 30 days after the event. In case you missed a lecture you were interested in, you can watch it again later.
Hello, good morning, good afternoon, and good evening to everyone joining our NutriTalks 2022 event today. My name is Nadine Heuer, and I will be your moderator for this long-awaited virtual training event, which I'm proud to say over 1,000 people have signed up for. We are looking forward to a very insightful and inspiring day with a lot of diverse speakers from around the globe dealing with the essential topic of nutrition. As already mentioned in our preface video, our agenda will be clustered in a total of 5 subject areas that I will guide you through step by step throughout the day. Each lecture will be paired with a question and answer session following afterwards, but limited to 10 minutes each, which you have the opportunity in to forward your questions and stress your remarks and thoughts to our KOLs. All questions posted during our event will be noted, provided with answers, and consequently uploaded onto our virtual platform by tomorrow evening for your kind review. Therefore, in case we do not get an answer, your question, one of our question and answer sessions, feedback will definitely be provided together with our experts for your review and to get hold of. The platform will be active for a total of 30 days. Of course, there will be times for break as well. We will have several integrated coffee breaks, including a 1-hour lunch break at noon CET, allowing you to have a stroll through the wealth of information provided in our lobby. For instance, with regards to the b.braun's represented nutritional products, information about our speakers, the Aesculap Academy, our NutriTalks event itself, and last but not least, to receive a certificate for your attendance by signing the respective form. One last thing to mention before we get started. Tomorrow, we are having another NutriTalks event with the exact same topics, starting at the same time as of today. However, compared of today, all sessions will be in reverse order to enable everyone around the globe to participate and to be included independent of different time zones. With this being said, let us now officially kick off our event with our first session block, dealing with pharmacy nutrition compounding. And let me herewith introduce our first speakers to you, which is Mr. Ikram Ali from Bradford Teaching Hospital NHS Foundation Trust in the United Kingdom. He is the lead pharmacist there and in charge of the quality management systems, mainly ensuring that GMP processes are followed. And secondly, Mr. Sebastian Wittenzellner, who is Senior Product Manager for Compounding at b.braun. He holds a degree in biochemical engineering, and his major focus in his current position is on how automated compounding devices can support pharmacists best and make the compounding processes safer. Both of them will share essential insights on safe admixtures of parenteral nutrition based on 2 b.braun products, which is namely EasyComp and Apex. Unfortunately, Mr. Ali will not be able to join personally today due to illness. However, you can place your questions on the lecture in the chat anyway. We will collect them and answer them afterwards. Nevertheless, starting with the first admixture tool, EasyComp, let us warmly welcome Mr. Ikram Ali, who will share his expertise on what EasyComp is all about. Video off.
Hi, my name is Ikram Ali. I'm a hospital pharmacist that currently works in Bradford Teaching Hospitals, and I will be doing this presentation on compatibility in compounding and experience with EasyComp software in a hospital setting. So hopefully in this presentation, we'll go through the agenda, which will outline some of the recent history of aseptic compounding and some of the errors that we've come across during our time with TPN compounding. We'll look at some of the regulations that are involved with electronic softwares, and we'll look at some of the challenges that are faced in technical services. And then we'll look at the EasyComp system and the compatibility software on that, and how we currently use it within our practice within Bradford Teaching Hospitals. And hopefully at the end we can have time for questions. So in regards to the background of who I am, so my name is obviously Ikram Ali. I'm a paediatric pharmacist and technical services pharmacist. My career, it started out in Newcastle upon Tyne, which is a tertiary hospital. I started as a rotational pharmacist and moved to becoming a paediatric pharmacist. And that's probably where my first insight into TPN actually began. I was working on a surgical ward, which had a lot of babies that would have surgical procedures and end up on short-term PN. And I also covered neonates there as well, which obviously a lot of those premature babies would be on TPN. I moved from Newcastle upon Tyne to Leeds Teaching Hospital, which was another tertiary hospital. And again, I was involved with neonatal intensive care unit and surgery in those babies. And again, those babies also required TPN. But this time round, I ended up moving into technical services, which deals with the compounding of the TPN and looking at that side of sort of the service. So in regards to that, I ended up looking at a lot of the software that we currently use, and I realized it was something that I was actually interested in and something that I would enjoy doing, and I loved doing the coding behind it and the software validations that was involved with that. I then in 2018 joined Bradford Teaching Hospitals as part of the technical services team as a full-time member there, and I would also cover the clinical pharmacy ward rounds for adult TPN and neonatal TPN, just due to my background in helping in ward rounds for TPN and attending MDTs for the TPN ward rounds. So that's who I am. So what we're going to look at now, hopefully, is looking at some of the errors that have been made within the pharmacy accepted unit and trying to see what we've actually learned from them and what did actually happen in those situations. So the first scenario that we have was to do with baby Alyssa Shin, which was a case from the USA in 2005. And in this situation, it was a 26-week gestation baby. Birth weight was 1 pound 4 ounces. And what had happened is they'd done a ward round and sent a prescription to the pharmacy team, which deals with the technical services part of it, or the compounding of that TPN. On inputting the prescription onto the computer software, which was a manual process, they added in the wrong dose of zinc. And this happened due to a number of reasons. One, the prescription had come down quite late. There was a difference in prescribing between how it was prescribed and how it was inputted. And the pharmacist had previously made an error, and so they were very nervous in regards to checking this prescription. Another factor that also was raised was the training of the team that was involved, and their lack of knowledge in regards to what was happening, and having a belief in the insurance of the pharmacist that everything was actually correct. So the quantity that was inputted into this software system was incorrect, and it was a lot higher than should have been. Unfortunately, the baby died due to a zinc overdose, and it was noted in this that there was multiple stages within the process where something could have been done and it wasn't, and there was a failure between each of the healthcare professionals that was involved. The next case happened in the UK, and that was baby Jada Pilkington, another 26-week gestation baby. And this time round, there was an overdose of glucose. Instead, the pharmacist put down 40% instead of 4%. In regards to that, it's probably like on a aseptic sort of process, sometimes like if 40% doesn't seem out of the ordinary, to be fair, because it just potentially could have been related to the child and the need for a smaller volume, so hence they used a higher glucose concentration for that. But this was to do with a prescription, which obviously they wouldn't have had a 40% bag. A lot of the babies on the neonates tend to get a central line, but obviously in this situation it caused a lot of catastrophic problems for this child. The glucose itself acts like an osmotodiuretic and causes damage to the veins, which obviously was not ideal, and caused death in the child. So from that, there was, and also other errors that had happened. There's been a lot of work in trying to gather a lot of the information in regards to errors that happened within hospital pharmacy compounding errors, because TPN is not the only thing that is actually made. There's a number of things that are made within the compounding units, such as cytotoxics and aseptic products and CVAS products. So there's a wide range of products that are made. So the NARES reporting system is a system which collects data from hospital compounding units throughout the UK, and this tends to include near misses that have happened. So early data that was collected between the period of 2004 to 2007 was analysed, and 43 hospitals contributed partially or fully during this time period. And that's an awful lot of hospitals, if you think about it, that have an aseptic unit. So it was a good collection of data. So in total, 4,691 reports were submitted, and an estimated 958,532 products were made with an error rate of 0.49%. And that probably, it doesn't seem like a high error rate, but you have to remember a lot of times errors are not reported as they should be. There's a lot of underreporting that tends to happen, and what we tend to see is only the tip of the iceberg. Some systems or some places or compounding units have a robust sort of quality management system, but again, this is dependent on the people that are reporting and making sure that everything has been reported correctly and it's got a clear process. However, that's what was reported. So 24 of these errors that were detected were during or after administration. So that's 24 potential chances of having a catastrophic sort of error that has happened. So a vast proportion of the errors that did happen were related to labelling, and that was 34.2% of the errors, which is an awful amount, actually. However, during this time period, a lot of the electronic systems were probably not in use. So a lot of the processes were probably manual and probably more at risk of having a human error involved in them. So in this table, there is all the errors that were associated with the preparations of PN in hospital pharmacies. So there's two columns here. So one is for the adult parental nutrition and the pediatric parental nutrition. So it lists the types of errors that were identified, and so you can see a vast proportion of the errors that have happened are to do with the labelling. So 34.2% of the errors that happened were to do with the label, and the other percentage of errors that has happened is due to transcription. Both of these are most likely to do with human errors. Some of the electronic systems, or most of the electronic systems, will be able to do the correct calculation, identify the correct drug, the strength, etc., because it's built into the system. Whereas labeling and transcription are almost linked between the two in regards to the processing of the work that's been put into there. Errors associated with the preparation of the PN. So it looked at the different areas in where the error was identified and tried to help identify what stage it does tend to happen. So it looked at the different stages of where the errors did tend to happen. And if you look very closely, a lot of the errors tended to be detected at the first check in assembly area for the adult TPNs, and the same for the neonatal or the pediatric CPN patients. So it shows that there's something before that process where something has affected that critical pathway, and there's something that's affecting that and causing the errors to actually occur. So whether it was the system, whether it was the operator, whether it was the workload, or the other factors that are involved in that, or not having like a robust system in doing the validations, was another thing. A lot of the sort of detection was spotted on the final check. So it shows that there's been a massive sort of slipping of a report of identifying the error before it's actually got to there. And that could potentially be because some of the people that are compounding don't tend to see the request, and they only see the worksheet in regard to the preparation. But somewhere along the line, it means that more people have missed this error, and there's a risk that it's going to bypass the final checker. And especially in the neonatal or the pediatric TPN, the error there is quite high. It's 26.3%, and those are the patients that would potentially worry more about, because they're the ones that are the premature babies that are low in birth weight, that have multiple sort of organ issues related to them, or they have like a weak gut and have like a low immune system. So they're the ones that you don't want to make an error, as you don't want to do with the adults either. But the greater risk is for the neonatal TPN or the pediatric TPN, because they're more vulnerable. So looking at the outcomes in regards to those errors that were reported, there was a vast proportion of those had minor issues related to them, but there was some, as you can see, for the neonatal or pediatric, that was a high percentage, 11.7%, which is obviously a high number, and then obviously the same for the adults, which was 15 cases that reported to 2.2%, and that's far too many, really. So it shows that there has to be something that can be done, whether it's introducing new systems, new software compatibility data, having new processes in there that will help identify why these errors are happening, or whether it's just to do with training. But trying to simplify the process is sometimes the best way to do it. So to summarize, labeling, the transcription errors accounted for the vast majority of the errors, so 45%, and 23.4% of the pediatric PN errors were classified as moderate to catastrophic patient outcomes, and the study is representative of 43 hospitals. So the study relies on detection and self-reporting of errors by staff working within these units. But as we've mentioned, sometimes that reporting isn't as it should be, and there's a mass lack of reporting at times, potentially because people may feel like it's more paperwork, or the issue has already been identified. So adding more reports to this may not be beneficial. So more recent data shows that more reports have been submitted within an 18-month period, and labelling worksheet generation errors account for the vast majority of the errors that do still occur. So 28% for labelling generation and 22% for worksheet generation, which is again a large number. So it relies on almost the releasing pharmacist or the releasing individual to be able to identify those issues on final check. And even though a small percentage of those errors were identified during or after infusion, which is perhaps again could be underreported. So if you just take a moment to think about your current PN prescription to manufacture process, what do you currently do in your own practice? Do you currently use handwritten or electronic prescription sheets? Are these sent down via fax or via email? And what do you do in regards to stability? Do you have a system that does the compatibility check for you? Do you have a crib sheet that does the compatibility data, or do you call the actual company to see if they would provide you with a stability data? And also, does your software allow you to generate the worksheets and the labels from the same system? And does it allow you to process a prescription, even if there's a warning on there in regards to the dosage or the safety of that prescription? I have looked at a number of software systems where sometimes it doesn't show you the warning of the dosage, which is quite dangerous, and it shows that there are systems still out there that don't actually do that. But it just does show you that, that there are other systems that people would potentially be using that could cause errors. And can you think of any near misses or never events in your aseptic unit where the errors could have potentially led to patient harm? I've had one situation that springs to mind where we had the wrong amount of sodium that was added into a TPN bag, and it had been because somebody had switched or added an ingredient onto the system and removed a different ingredient onto the system, which could have led to potentially a lot of errors had it not been identified, and patient harm as well. At the end of the day, that's the one thing that you want to care about, and that's patient-centered care and making sure that the patient's getting the best out of their treatment and there's no risk to that patient. And looking at the deviation or the management of that, we'd seen that it's been somebody that hadn't used the system in quite a long time. They were trying to get something to the best fit, and they hadn't actually reread procedures. They hadn't done any of the training. They hadn't cascaded any of the information down, and they'd done it by pure accident, but it still caused an issue, and it could have caused catastrophic harm to the child. So where do we get our standards from? So my unit currently within Bradford Teaching Hospital is a Section 10 unit, but it also has a license for batch preparations. So the preparations of most of the medicines within the NHS are separated into these two categories. So one under a medicines and healthcare products regulatory agency license, or under the Section 10 exemption. And for the Section 10, which is what we normally work with in regards to the TPN, hospitals must comply with the defined standards, and they are inspected against these regularly by the regional QA specialists who will come every year and ensure that you've done everything by the book, and you still are doing everything by the book. And if there is any errors, etc., they are clearly identified and clearly investigated with appropriate actions to be done on there. So just a quick overview of some of the standards. So this is just from QAPS, which is our regulatory information for Section 10, quality assurance or accepted preparations. And it mentions just here that the computerized systems that we use, the software systems that we should use, should be a validated system, and it should be risk-based approach to decide the level of validation that's required. And it should demonstrate a level of accuracy and traceability which is at least as good as any paper-based systems. And that links to the Aspen guidelines, which says that parental nutrition should be prescribed using the standardized PN order format, and the review processes should be applicable to patients of every age and disease within the healthcare organisation. And it mentions about computer systems and handwritten orders and prescribing TPN and potential for errors, and making sure that we avoid any sort of verbal and telephone orders for PN. And it kind of shows the clinical decision support should be available within electronic PN orders to alert and prevent prescribers from ordering doses of macronutrients and micronutrients, and making sure that they're aware of what the clinical limitations are and whether they exceed those limits of compatibility. So one of the recommendations from looking at TPN by the chief pharmacist was to ensure that the development and implementation of an action plan to eliminate handwritten documentation and minimize the use of pre-printed and photocopied worksheets was in place. So it shows, obviously, if we take it back to the errors that we had, it was to do with transcription and labeling, which in theory, if you had an electronic system that was linked to your server, that linked all the data to your patients, that error would be minimized because it's been coming from the server, and it would drastically reduce those errors. And the chief pharmacist basically said they must ensure that the software used in the accepted unit to produce documentation and labels is validated according to standards specified within the guide to good manufacturing practice. So we'll come on to that now, I think. So what happened with us in regards to Easycomp is we had a system that we used, which was NutriFlex, which was also a b.braun system. When I came to the trust, I'd realised it was a very old system. It wasn't up to date, and the license on that had actually finished and had been updated to the Easycomp system. So the validation paperwork that we had was quite out of date. The compatibility data that was previously there wasn't available, and we came to a point also where the bads that we used, or the bads that we were going to be using, were not on the NutriFlex system. And to be honest, it'd been in place for that long. A majority of the people that had been there did not actually know how to add new ingredients onto the system, and they did not know how to validate the paperwork either. So we knew at that point that we would have to use a new system. And we basically did what it said in QAPs, and we did a risk assessment, and we looked at the risk assessment and said, well, what systems are there available and what systems are being used currently in different trusts within our region, to see if we could help standardize things that we were doing. And EasyComp was one of the systems that was currently there. I won't talk about the other systems that are present. I'll just focus on EasyComp at the moment. So we did a risk assessment, and we looked at how we would approach it. And so we looked at how it was validated from the supplier itself, and we looked at the essential functions of the software, but we did a proper validation in the sense we looked at the user requirement specification. So what did we actually want from the system? What did the regulations want from a system? And then we processed the whole validation based on that. And so we did our design qualification, we did our installation, operational qualification, and performance qualification based on that. And we looked at basically the essential functions that it should have, which was part of the MOSCA requirements of what should be present in an electronic system, high priority sort of must-haves, but those that were not essential and could have, which were the desirable but not necessary, and the ones that we would like to have in regards to functionality of the software. So we switched to EasyComp system, and the validation was undertaken in 2019. It was a relatively quick validation. The software had been used by a number of sites within the region, and the software was fairly easy to use, and it just allowed it to easily kind of go through the system. We had a lot of support from our IT support, and we had a lot of support from b.braun at the time, which was invaluable because obviously they knew the system, and we kind of did the validation with some oversight in knowing how to use the system from them. They did a lot of training that was there. So in 2021, we had about 60 patients overall in that year that had TPN. So we prepared about 1,200 bags in our aseptic unit. The way that our TPN system works is all referrals go through the TPN team. It has to be seen by a dietitian, and they have to then decide the TPN content that they would like for that patient. We've tried to standardize a lot of our TPN and try to make it more simpler and more smoother, just to allow for contingency and capacity, and at the same time ensure that the patients are getting the right amounts in their TPN. So the benefits of the system that we noticed straight away was the patient data archiving system that was available. There was a clear traceability of who had been involved in what process. There was an ease of processing. So I'm not sure if people had used the previous system, but it was difficult to kind of see at what stage anything had happened. It was a massive list of patients and processes that you had to get to that point. And there was an inbuilt compatibility data that was based on the stability matrix, and that looked at both the physical and the chemical data. So one thing that did happen is we used to have a system update on a regular basis, and I can't actually remember how often it was, but it switched now to a quarterly basis. One thing perhaps to say with that is it did cause a lot of work to be involved, a lot of planning. It would require somebody from b.braun, it would require someone from IT, and then it would require a lot of work from ourselves in regards to the revalidation, because once it's been updated, you have to revalidate the entire system again, just to make sure that there's that assurance, to make sure nothing has gone untoward or nothing has corrupted in regards to a system bug, which potentially could happen with any system. So it was just making sure that we'd done all the relevant checks that we had done. And I think it changed after a while because we realized how much work was going into that. We kind of asked people on, could we change this to like quarterly update, which did happen. And it seems to work a lot better for us in regards to that. So in regards to the features that we did want, in regards to the electronic compatibility software system, we looked at the things that we would need to do. So the IT requirements. So what did IT need to know? So did it require a Windows 7 system? Did it require networking to our software and to bypass some of the trust firewalls? Was there compatibility in regards to the computers that we used and the labels that we used, and like the trust port compatibility? We looked at the security and passwords and just tried to see whether it allowed us to give different levels of user functionality to people, and how often it would change passwords, etc. We did like a general overview of how the system works, how it looks at configuration of ingredients that you use for the macro ingredients and the micro ingredients, and the TPN choices that you have in regards to base bags as well. We again looked at worksheets and labels and made sure that all the data that has been selected was populated, whether it would match the regulatory sort of requirements that we need for worksheets and for labels, and whether there was any scope to configure that to what we wanted on our worksheets and our labels. And just because we'd always had certain things that were there, and there was regulatory requirements that we need to fulfill as well. And just configuration and system contingency was obviously an important thing, because and life cycle, actually, because for our previous system, that's what kind of had let us down in regards to maintenance of that system and revalidation, and making sure that we had another system already available to us that we could use, especially if a system goes down, or if there's not going to be a system that's going to be used anymore if it comes off cycle. So they're all very important things, and we processed them throughout the validation of our software paperwork. One of the key features that we obviously identified straight away is obviously the software that was available for the compatibility of EasyComp, and we didn't really have that before with our other system. It wasn't up to date, it was out of date, and it was just so much easier with this system. It would let you select the electrolytes that you were using in the actual TPN system, so you could check what was going into the compounding of that bag, and it would flag up any sort of issues that might have been identified, whether that be it different ingredients that should not be used together, or whether it was high quantities of one ingredient and low quantities of another. It would tell you what was going on. And you could also have an option to use it to set your own expiry dates on the system. So we set ours as 4 days at room temp and 2 days in fridge, because that's what that's the minimum expiry we would need, and that's the maximum we would want to give to our TPNs. I think that was everything that I had to add in regards to the EasyComp system and the software compatibility data and how we use it within our trust. But if you have any questions, I'd love to hear them. So thank you for your time, and thank you for listening to me.
Well, thanks to Mr. Ali for sharing his expertise with us despite of not being able to be with us today. We are now proceeding with our subject area, pharmacy nutrition compounding, but this time with the example of learning all about Apex. Sebastian, the stage is yours.
Welcome to our today's NutriTalks. I am Sebastian Wittenzellner, Global Product Manager for Compounding at b.braun, and I'm proud to present to you today Apex, our new automated compounding device for parenteral nutrition. In this session, we will have a look at the tasks and challenges in the pharmacy and how Apex can make the process of nutrition compounding safer and more efficient. After this session, will be a live question and answers for your questions. Feel free to write your questions during the lecture in the chat and I will answer them later. The task of the pharmacist is the safe preparation of medication. This preparation must contain the correct substance in the right quantity at the required purity in the correct container with the correct label. This requires accurate work in all steps in the preparation process, from ordering, quality control of incoming goods, over preparation, validation, and documentation, and many more. Pharmacists are responsible to fulfill all these requirements. Parenteral nutrition regimens are complex sterile mixtures of a large number of macro and micronutrients in various combinations. They range from volumes of 100 ml up to 4000 ml for adults. They are available as ready-to-use formulas like 2 or 3 chamber packs, or, in our case, patient-specific preparations for patients like preterms and neonates, patients suffering from renal failure, or oncology patients, where requirements might change on a daily basis. They can be compounded either manually or automatically. Challenges for the pharmacist include safety concerns related to solution identity, dosage, contamination, compatibility, and connectivity. Time can be an issue depending on the number of prescriptions per day, setup time for automated compounders, and time-consuming documentation. In addition, the pharmacist must weigh the cost of the equipment and the cost of qualified personnel required. Let's have a look at the Apex compounding system, the new state-of-the-art compounder for total parenteral nutrition. It comes with a 26-port transfer set, which is pre-assembled, a vial rack for the handy accommodation of ingredients directly at the compounder, a large, flexible 16-inch screen increases legibility and can be placed either in front of the compounder or anywhere else in the laminar airflow, a barcode scanner, reduces cable clutter in the workspace and allows for patient and solution verification without human error. And Apex has a load cell for gravimetric verification of the compounded bag. This is adaptable for horizontal or vertical airflows. And finally, Apex is the only compounder with a dual drive, a filling technology for precise macro and micro delivery at the same time to speed up the compounding process. Safe and efficient compounding with Apex. b.braun offers a complete package for the compounding process, from order entry, where Apex can process PAT files from many different sources or read barcodes, from entry via prescription software, error-free transcription, checking the compatibility, dedicated in the complete portfolio of disposables, accurate and efficient preparation with Apex, the precise delivery via infusion pumps, to documentation where various types of reports are available. The process of parenteral nutrition starts with a prescription. And one of the main risks are transfer errors of the composition of the regimen from the label to the mixture. Apex can read and process prescriptions from several established prescription software in the market. The prescriptions are sent via local networks as so-called PAT files to Apex, or the software like b.braun's cleaners are printing barcodes of the label, which can be read by Apex. This simplifies and optimizes the prescription of clinical nutrition and offers a closed loop from prescription to administration and helps to reduce medication errors by a human error-free transcription. Another very critical point for patient safety is compatibility. Parenteral nutrition regimens are complex admixtures and it is difficult to predict whether the prescribed regimens will be compatible or not. Since the prescriptions are according to the patient needs, it is the responsibility of the pharmacist to make sure that they can be mixed safely and administered safely. For example, the excessive calcium and phosphate in the TPN solution will form a precipitate, which can lead to pulmonary emboli, respiratory distress, and even death. EasyComp is a GMT-validated compatibility program and allows the pharmacist to check whether the chosen nutrition regimen will be physico-chemically stable or not, and if yes, for how long. EasyComp has a server-based database which refers to thousands of laboratory tests for its compatibility statement. If the regimen is outside tested nutrition ranges, EasyComp allows to modify the regimen to put it into the window of compatibility. If compatibility is confirmed by EasyComp, it provides a stability statement and shelf life, prints labels, worksheets, and prescription sheets if necessary. Consumables. Apex is used with a pre-assembled transfer set that allows a fast and easy setup. 26 ports maximizes the number of ingredients which can be delivered automatically and minimizes the number of ingredients which have to be added manually. The set is color-coded, numbered, and barcoded to avoid mix-ups and to make the setup easier. The barcode avoids the use of the wrong solution by scanning the barcode of the solution and the line. Since the set is pre-assembled, the risk of contamination is reduced. The set comes with 7 flex lines, which allow dispensing of the solution from the same container either via the macro or micro line. This depends on the volume which shall be dispensed. For volumes less than 10 ml, the micro line is used. For larger volumes, the macro line is used, which is up to 6 times faster. A broad portfolio of bags is available with Apex. Standard EVA bags with volumes from 250 up to 4000 ml. Dual chamber bags where lipid solutions shall be separated from the rest of the regimens are also available in various sizes, starting from 1500 up to 4000 ml. On top, b.braun can offer multi-layer bags which offer an excellent oxygen barrier and reduce the UV light transmission. The degradation of vitamin C as one of the most sensitive vitamins is almost completely eliminated after a few days. This protects the regimen and increases the shelf life. The vial rack and the easy clip vial holders allows an easy and comfortable accommodation of the small volume containers directly at the compounder. The connectors to connect the channels with the containers are not pre-assembled. They have to be chosen according to the container. Mini spike for vials, fluid dispensing connectors for syringes, and for large bottles and bags we offer the universal spike. Compounding errors can be incorrect dosing, wrong concentration, or mixing up look-alike containers of source solutions. The barcode scanner, which is wireless, allows to verify the right container at the right station and is used for the order confirmation on the label at the back. This helps again to reduce human errors. On top, the barcode scanner is wireless and reduces the cable clutter in the workspace. Apex has a large display of 16-inch, which allows easy activation of the different buttons and gives a clear overview of the compounding process. The screen is structured in multiple areas, which are clearly separated from each other. On the left, the flex lines, which can be used either for the macro or micro delivery. In the middle, dedicated micro channels, and on the right, dedicated macro solutions. The display is flexible and can be placed either in front of the compounder or anywhere else in the laminar airflow. All solutions which are on the label but not hanging directly at the compounder are automatically listed under manual additions. This increases safety because no manual addition can get lost anymore because a solution was not hanging directly at the compounder. Apex has a unique dual drive that can deliver macro and micro solutions at the same time. This helps to speed up the compounding process whilst maintaining the accuracy. Now, let's have a look at the complete process in a short video. I hope that this short video showed you how efficient the compounding process with Apex can be. Now, last but not least, Apex offers a variety of reports for the pharmacist. This can be the authorization report, the compounder event report, the compounding activity report or compounding precision report, the final container usage report, the solution usage report, which is selectable by day, week or year, and the solution usage report. As you have seen during this presentation, b.braun wants to support the pharmacist with a complete package for the compounding process. With Apex as state-of-the-art compounder, excellent service, a new prescription software, compatibility software, and nutrition solutions, we cover the complete process of parenteral nutrition, from the prescription to administration for more safety in nutritional therapy. Thank you very much for your attention. I'm now available to answer your questions in the live Q&A.
Thank you very much, Sebastian, for this great presentation, and welcome to our first live question and answer session. Sebastian, are you actually ready to get challenged?
For sure, Nadine.
That's great to hear. And we already have a couple of questions coming in, and
Okay,
I would
good.
like to ask you the first one. So, what kind of single nutrients and solutions like amino acids can be used with your compounder, and do we have to use b.braun products for that?
Okay, that's a good question. Of course, it would be great for b.braun if only our products were used for compounding. But to offer our customers the greatest possible flexibility, nutrition solutions from other manufacturers can also be used. Together with our customers, we take a close look at which kind of solutions should be used. And based on this, we are developing together a so-called delivery factors. Based on this solution database, these individual parameters are then later used for the exact pumping of the machine.
All right. That's good to hear. And the next question actually goes into a little bit into the same direction. Can we also use other bags from other companies with the compounder?
Okay. Apex in this case can only be used with our dedicated b.braun disposables, and this is the only way for us to guarantee the accuracy of the machine. Remember, as you have seen, Apex offers an accuracy of ±3%, which is among the highest in the market. And this can only be done with own dedicated disposables.
All right, so let's summarize. We can use the solutions of other companies, but let's say the disposables are only dedicated to the b.braun portfolio. All right.
That's correct, Nadine.
And there's another question with regards to multi-chamber bags and compounded bags. So the question is, isn't it recommended to use multi-chamber bags instead of compounded bags? And what is actually the benefit of having compounded bags? Thanks.
Okay. This is correct. The use of multi-chamber bags is recommended for many indications, and both kinds of nutrition therapy have their own benefits. So, for example, for compounding, it's often used whenever individual patient needs change quickly and frequently. And this can be the case, for example, with premature babies or cancer patients.
All right.
So this
Good.
always depends a little bit on the situation.
Thank you. And we also have a question with regards to the stability. So once the solution is being compounded, so how long is the PN solution in bag stable?
For this, we offer the EasyCom software, for example. We have done several thousands of tests with regard to the compatibility of the different kind of solutions. And EasyCom offers the possibility to get a statement at the end, either if the composition is stable or not, and if it's stable, for how long it's stable. And in general, it can be said that the stability data is valid for 7 days in the fridge, plus another 2 days out of the fridge.
Okay, so it's actually recommended to use the EasyCom software in order to
Yes.
get the right data for that. Okay, great.
Exactly.
And we also have a question with regards to the barcode. What is actually the role of the barcodes at each line?
Yeah, the barcode helps to reduce human errors during the compounding process. So the step one would be to scan the barcode on the solution, on the container. There's a barcode on the container, and this is then linked to the channel of the compounder. And if there's a mix-up, let's make an example. So you want to use an amino acid. Let's use a b.braun solution, amino plus mal 10%. And by accident, you're scanning a quite similar solution, amino plus mal 10% HEPA. Then the compounder will recognize this error, avoid any proceeding, and ask the user to correct the error. So it can be make sure that the right container, the right solution is used with the right channel of the compounder to really assure the accuracy and compatibility of the compounded bag at the end.
I see. Okay. So let me quickly check on the chat if we have any further questions. But so far, I see no further questions. But, just feel free to post further questions if they come later to your mind. It's really no problem to send them via email or to send them via chat. We will collect them and answer all of your questions even after the event. So Thank you, Sebastian, for this great presentation, and welcome to really this wonderful question and answer session. That was really nice. And also to the audience, if you would like to learn more about Apex and EasyCom, you can visit the booth in the exhibition area to find more material information on it. The two of us and the audience will now take another short minute break to refill your cups, and I think we'll see you at 10:40, yes, I believe it's 10:10. Hello, everyone. It's 10:40 CT, meaning time to proceed with the subject area number 2 of today, dealing with the topic nutrition in ICU. Within this area, we look forward to welcoming 2 speakers, namely Professor Dr. Adolf from the University Clinic in Tübingen, as well as Dr. Norbert Nagel from our b.braun Medical Scientific Affairs Department. Dr. Nagel has a degree in biochemistry and brings with him a wealth of knowledge and more than 15 years of experience in the area of clinical nutrition. He is currently in charge of the clinical nutrition area as a global senior medical scientific affairs manager at b.braun. But first in line will be Professor Dr. Adolf, who is anesthesiologist and ICU physician at the University Hospital of Tübingen, doctor of the first hour. He has substantially contributed to the establishment of clinical nutrition in Germany by establishing a fully operating professional team of dietitians and nutritionists at his hospital. He will give us now an insightful speech on the benefits of omega-3 fatty acids in total parenteral nutrition with a specific focus on Lipo+ and lipidem. Professor Dr. Adolf, the stage is yours.
Dear ladies and gentlemen, dear colleague, it's really a great honor for me to be with you today in this part of NutriTalks. My topic today is the benefits of omega-3 fatty acids in TPN, and actually I'm focusing on the product of Lipo+ lipidem. My working place is the University Clinic of Tübingen. Tübingen is an old university town in the southwest of Germany. On the right side, you can see a picture out of my ICU ward. It's a great ICU ward with 45 beds. During the last 2 years, we had a lot of work with the COVID ARDS patients. We see one of them here in this picture. Actually, I want to discuss with you the metabolism and the nutrition support discussion around these kind of patients. What happens in critically ill patients? We know for many years that we have the problems of inflammation. According to the multiple trauma injury, burn injury, hemorrhagic shock, the organism is triggering the inflammatory response. On the other side, bacteria, viruses, fungi, anaerobes might also trigger a septic condition. Either sepsis or inflammation is going down to a secondary organ damage and will end up in a complete organ dysfunction. And more or less every organ can be affected. That means the brain, lungs, liver, kidney, cardiocirculatory system, GI tract, immune system, circulation. And this in the end is really a critical ill patient. What is the best nutrition strategy in critical ill patient? We can follow this circle. First, we have to do a nutrition risk assessment. We have to identify the highest risk patients using special tools. Then the amount of nutrition is important. We can use, if available, the technique of indirect calorimetry to get a clear signal what is the energy consumption. Then the route of nutrition is important. Should we use enteral? Should we use parenteral? Or should we go with a combination of both, which is called supplemental parenteral nutrition? I feel this is the best way for most of the critical ill patients. Then the energy intake is, of course, very important. We want to go with a balanced intake of fat and carbohydrate energy. And today we are focusing on the special substrate of omega-3 fatty acids. In the end, we have to monitor our nutrition regimen. That means we have to do a daily reassessment. We have to take laboratory data, and we have to check what happens with the glucose. That means we have to do a good glucose control in our critical ill patients. In the next few minutes, I will focus a little bit on the international and national guidelines. And the question for all of us is there a consensus or is there a conflict between these guidelines? We have the German guidelines, we have the European guidelines, and more or less very new are the guidelines from the American Society of Parental Enteral Nutrition. The Aspen came up with these recommendations in adult critical ill patients receiving PN, thus provision of mixed oil intravenous lipids, that means medium chain triglycerides, olive oil, fish oil, or the mixtures, as compared with 100% soybean oil, impact clinical outcomes. And they concluded, they recommended, owing to limited statistical or clinical significant differences in key outcomes, we suggest that either mixed oil intravenous lipid emulsions or 100% soybean oil containing lipid emulsions be provided to critical ill patients. And there I will definitely put a question mark, because this is, from my point of view, and from the point of all the experts here in Europe, this is wrong. We should not longer use the 100% soybean oils. And the next question was, in adult critical ill patients receiving PM, does provision of fish oil containing lipid emulsions as compared with non-fish oil containing lipid emulsions impact clinical outcomes? And they recommended, because there was only one outcome with a significant difference that was not supported by data covering the other key downstream outcomes, we suggest that either fish oil or non-fish oil containing lipid emulsions be provided to critical ill patients. Again, there must be put a question mark, because we got a lot of papers, we got a lot of good, excellent research showing the benefits of mixed lipid emulsions containing fish oil. The German guidelines, we recommended fat should be an integral part of enteral and parental nutrition. In critical ill patients, parental nutrition shall include fat emulsions containing reduced amounts of omega-6 fatty acids. That means don't use the old type, the first generation of lipid emulsions containing only soybean oil. And we recommended patients should receive parental fat according to the phase of the disease and individual metabolic tolerance. Medical nutrition therapy should respect a maximum of 1.5 gram fat per kg body weight per day. And during parental nutrition, patients should receive a continuous infusion of fat emulsions in a time period between 12 and 34 hours. Please don't use a bolus application. European guidelines, they recommended intravenous lipid, including non-nutritional lipid sources, should not exceed 1.5 gram lipids per kg body weight per day and should be adapted to individual tolerance. This is also very important. And with regard to the intake of omega-3, they recommended parental lipid emulsions enriched with EPA and DHA. The fish oil dose should be in between 0.1 and 0.2 grams per kg body weight per day, can be provided in patients receiving PN. When we take this dosage recommendation, it's important now to check whether the product will deliver this amount. We have two different European pharmacopoeias focusing on this. The EP number 1352 is telling us some of the contents of the omega-3 acids, EPA and DHA, expressed as triglycerides. There should be a minimum of 45%. And total omega-3 acids expressed as triglycerides, there should be a minimum of 60%. And following this, the highest EPA and DHA and total omega-3 acids content can be found in the LipoPlus lipidane product. And this LipoPlus lipid emulsion is also part of the convenience system called the NutriFlex Omega. Most of the other commercially available lipid emulsions containing fish oil on the market are following the EP number 1912. This is shown on the right side of this table. And this comes in general with definitely lower amounts of EPA and DHA. Keep in mind, please. The Aspen also made up a few years ago an expert group for lipids. I was part of this group, and we concluded at that time, fish oil enriched enteral and parental nutrition appears to be well tolerated who confers additional clinical benefits, particularly in surgical ICU patients, due to its anti-inflammatory and immune-modulating effects. Whilst the evidence base is not conclusive, there appears to be a potential for fish oil-enriched nutrition, particularly administered perioperatively, to reduce the rate of complications and ICU and hospital stay in surgical ICU patients, as well as to improve complications such as intestinal failure, associated liver disease, associated with the old lipid emulsion containing only soybean-based. So the next step, let us go into more detail. I mentioned just in the beginning of my talk the inflammatory response. We have invading microbes, viruses. We have the injury, the tissue injury. And that means there is a huge vascular response. Special eicosanoid may be synthesized in a strong manner. And this will end up in a strong, very heavy inflammatory reaction. And our work is now, can we control this inflammatory response? Can we downregulate this response? Yes, we can do this by using omega-3 polyunsaturated fatty acids, because from these fatty acids, the organism is able to synthesize specialized process-solving mediators like the resolvins, the protectins, and the myrcins. We want to get in this situation a resolution of inflammation and hopefully coming back to a restitution of a normal structure to homeostasis. We know today that coming from EPA and DHA, we get the resolvins E1, D1, and D2. E1, for example, means the microbial clearance will increase. It's much better. D1 means nitric oxide and prostacyclin synthesis will increase. And D2, migration and IL-12 production is going down slowly. This is, from a clinical point of view, very good. But the next question is, this is a little bit of hypothesis. Does it really work? Yes. I show you only two different works done in a marine model. The first one was done to investigate the acute respiratory distress syndrome in a mice model. Hecker and co-worker investigated the effects of lipid emulsions on lung pathology and inflammation after induction of ARDS. The mice were infused sodium chloride, long-chain triglycerides, or a physical mixture of LCT-MCT or LCT-MCT and fish oil over 3 days, followed by an intratracheal application of LPS 24 hours prior to being sacrificed. Lung morphology was assessed by histology to evaluate the inflammatory effect. Before challenge, lungs of mice from all treatment groups displayed a similar histological pattern. After LPS installation, lungs of animals infused with sodium chloride showed a marked increase in the leukocyte invasion into the alveolar space and the interstitial edema as signs of pulmonary inflammation and damage. These morphological features of acute lung injury were even more pronounced in the LCT and in the LCT-MCT group. In contrast, leukocyte invasion and edema formation were ameliorated in mice receiving the mixture of LCT, MCT, and fish oil. So again, this is a perfect result. And they concluded fish oil containing lipid emulsions display anti-inflammatory and pro-resolving properties after LPS stimulation. Then the next step was done in a sepsis model. This was done in my working group here in Tübingen at the University Clinic. We used the technique of cecal ligation. Here we demonstrate that the omega-3 lipid emulsions orchestrate inflammation, resolution, regeneration mechanism during steroid peritonitis in a murine polymicrobial sepsis. Omega-3 lipid emulsions recessed neutrophil infiltration, reduced pro-inflammatory mediators, reduced the classical monocyte and enhanced the non-classical monocyte macrophages recruitment, and finally increased the erythrocytosis in sepsis. So we concluded omega-3 lipid emulsions lead to an improved survival during marine sepsis. This is not shown in this graphic presentation, but I can clearly argue that there was a better result in the survival rate in this marine model. And this is showing what is the theory behind the story. We have a normal tissue when we have the invasion of microbes and viruses coming with a neutrophil infiltration, and then hopefully the specialized process-dissolving mediators can do the counter reaction or counterbalance, let me say, coming back to this resolution, regeneration, that means TGF-1 beta and IL-10 will increase again, and hopefully we come back into this situation with a normal tissue. So omega-3 lipid emulsions play an important role during inflammation as well as infection and stimulate inflammatory resolution and tissue regeneration. Again, a picture out of my intensive care unit. It's showing you the COVID ARDS unit. It was a very strong work over the last 2 years, and again, we still have these kind of patients in our ICU ward today. Is there any clinical evidence for this? This is, from my point of view, really the most important question now. Let me say yes, some examples. First, we focus on cardiac surgery. This is a paper done in 32 infants undergoing open heart surgery with cardiopulmonary bypass. They were randomized to receive an intravenous lipid emulsions only with soybean oil. That means here on the left side, or the new mixture with medium chain, long chain, and fish oil. And in both groups, they got these lipid emulsions pre and post surgery. The intake was always the same. The energy intake, the total protein, total fat, total fluid intake was totally equivalent in both groups, in the treatment and in the control group. So what we found, or what they found at that time, infants who received an intravenous lipid emulsion containing EPA, DHA, and MCT, the black bars, before and after open heart surgery, had reduced plasma TNF-alpha concentrations compared to infants who received the standard soybean oil lipid emulsion. In general, there was a reduction in inflammatory cytokines with variable effects on anti-inflammatory cytokines. Next part, ARDS again. This is a paper from the Spanish group. They checked this new lipid emulsion coming with fish oil in ARDS patients, and they checked the eicosanoid synthesis. Thromboxane B2, LTB4, 6-keto PGF1 alpha. If you go with the fish oil, you will see a reduction in all these eicosanoids. But if you go with LCT, there's an increase. It was significant, and from a clinical point of view, it's very important. Septic patient. This is, let me say, a newspaper message. Adding fish oil to IV may speed sepsis recovery. This newspaper message came directly following to this study results from the Phil Calder group in the UK. They checked 25 consecutive patients diagnosed with a septic syndrome. Again, there is the comparison between the fish oil, the MCT, LCT fish oil, of course, and they checked it against the second generation, that means MCT and LCT. So from my point of view as an intensivist, the ratio of PaO2 and FiO2 is really very important. very important. And you can see after day 6, this ratio is much better in the fish oil group compared to the MCT-LCT group. It was statistically significant. And in addition, the length of hospital stay was much shorter in the MCT-LCT and fish oil group with 22 days compared to the MCT-LCT group with 55 days, and there was a p-value of 0.05. We stay a little bit longer with the sepsis discussion. This is a paper from Hall and co-workers. They were interested in the survival parameters, and they checked the organ dysfunction and the inflammatory response in general. Using the SOFA score, they could show a significant reduction in new organ dysfunction. There was a p-value of 0.005. And the general inflammatory marker of CRP was definitely lower in the fish oil group, and it was again statistically significant. Pancreatitis. In this study, Wang and co-workers compared again these different types of lipid emulsions. These were 40 severe acute pancreatitis, parenteral nutrition for 5 days in the double-blind manner, so it was quite okay. The dosage was quite okay. They used 1 gram of fat per kg body weight per day. And what we could show that the CRP levels were again lower in the fish oil group, and using continuous renal replacement therapy, the number of days on CRRT was definitely lower in the fish oil group, 18 days compared to 26 days in the control group. Again, a clinical benefit, of course. Critical ill patients. There's a very important study from Spain, though it was a multi-center trial. They were focusing on nosocomial infections in critical ill patients. And the main findings of the study is that administration of 0.1 gram of fish oil per kg body weight per day. You can remember the recommendation. It should be in between 0.1 and 0.2 in combination with MCT and LCT in the lipid emulsion significantly reduced the risk of nosocomial infections. You can see it in this graphic presentation. There was a risk reduction of over 40%. And the same study, the ICU lipid study, could also show that the time free of infection was quite different. It was statistically significant better in the fish oil group compared to the simple MCT LCT emulsion group. The question of the outcome. We got a lot of different papers over the last few years from Pradelli, Manzanares from Canada, Palmer, from China, again Manzanares. And there's a, let me see, really a new one, a good new one study, meta-analysis from Pradelli and co-workers. This meta-analysis is the largest to date investigating the effects of different intravenous PN regimens on clinical important outcomes in the ICU. So they differentiated between ICU patients and critical ill ICU patients. Critical ill ICU patient means that all of the patients were ventilated or needed some kind of organ support, or the ICU stay was a little bit longer compared to the ICU patients. But so focusing on the infection rate, they found by mixing different studies over the last few years, they could show that the infection rate was definitely lower in the fish oil-based PN groups. The asterisk is always on the left side. That means favoring the use of fish oil. And the next parameter is the hospital length of stay. The same. The asterisk is always on the left side. using fish oil is, from that strongly recommended, good results. And the ICU length of stay, again, the asterisks are on the left side. So infection rate, hospital stay and ICU length of stay can be improved by using fish oil containing lipid emulsions. We can show that omega-3 fatty acid containing PN is associated with a positive impact on clinical outcomes for ICU patients, such as reductions in infection rates and the duration of hospital and ICU stay. But they made a very additional analysis focusing on the budget. Why that? Why? Because omega-3 fatty acid containing PN is normally a little bit higher in the costs. And this is due for all the markets. You can see here France, Germany, Italy, Spain, United Kingdom, and also the US market. The cost definitely a little bit higher in the fish oil containing group. You can see it from here, these green bars. But then they checked the infection and the hospital length of stay, and all these very important parameters, of course, were better compared to the standard PN group. So despite the fact that the costs of omega-3 fatty acid containing PN is a little bit higher just in the beginning, they could reduce the costs for additional diagnostic procedures, additional treatment procedures. So in the end of the day, they could reduce the hospital costs and they could do something very positive for the budget. The best result could be demonstrated in the US market, but in all other European markets, you can see that there is a positive effect on the hospital budget by using omega-3 fatty acid containing lipid emulsions. So a pharmacoeconomic simulation revealed that omega-3 fatty acids containing PN is likely a dominant alternative to standard PN from a hospital's point of view for all of the 5 European countries analyzed, as well as for the US market. Despite higher acquisition costs for omega-3 fatty acid containing PN, its superior efficacy makes it a cost-saving alternative. What is the best strategy? We know that in critical ill patients, we have different time slots. We have acute phase, we have a post-acute phase. And in the acute phase, we have, again, 2 different time steps. We have an early phase and a late phase. I just tried to show you that we have to control the inflammatory response in our critical ill patients. And if we would follow this idea, we must start with the intake of omega-3 fatty acids as early as we can. If we do not follow this idea, if we wait, then the inflammatory response is overwhelming the whole organism. And then the intake of omega-3 fatty acids might be too late, or it cannot work in this way of idea. So please start as early as you can with the intake of omega-3. And I showed you the recommendations. The total intake of omega-3 in PN should be up to 1.5 gram per kg body weight per day, including, of course, the non-nutritional lipid intake. Don't forget this. And we should go with intake of 0.1 to 0.2 gram of fish oil per kg body weight per day. You can do this with this product, with LipoPlus, coming with MCT, LCT, and omega-3. If you take this product, you can absolutely meet the recommendations. And this is also part of the commercially available 3-chambered bag, the NutriFlex Omega system. That's the best way you can do for your critical ill patients today. So far, my lecture. I hope that you have a lot of questions now so that we can discuss it in the next round. For the moment, I will say thank you very much for your attention.
Well, thank you so much, Dr. Adler, for this highly interesting presentation, and welcome to our question and answer session. Now let's have a look at some of the questions dedicated to you and the topic you have presented. We have a couple of questions coming in by email, so I would like to raise the first one. It's a very interesting one. Which patient group would you definitely recommend the usage of lipid emulsions with omega-3 fatty acids for?
Thanks. This is a very good, excellent question. We had this kind of discussion many years ago when we got the new modern lipid emulsions in the German market. And we discussed it for a long time, but actually nowadays we use the omega-3 containing lipid emulsions more or less daily in the routine and in every patient type. There is actually no differentiation at the moment. Of course, as you mentioned during your short welcome speech, I'm an intensivist, and of course I'm caring for really critical ill patients. We have a huge ICU ward with more than 45 beds. We have a lot of COVID ARDS patients, and every type we have liver transplant, we have pancreatic transplant, we have trauma patients. So I can tell you that we use omega-3 lipid emulsions in every type of patients. Actually, there is no differentiation. And even if the patient is discharged to the normal ward, or again, if the patient is discharged to his home and he needs home parenteral nutrition, the recommendation is the same. We use and we recommend for the patients in the normal ward, but also outside when he is going home, we recommend the use of omega-3 fatty acids in lipid emulsions in all of these patients. There is no difference at the moment.
Okay, so I see you've already implemented that as a standard therapy already. Great.
That's correct. That's correct.
When would you actually recommend to start with parenteral nutrition, including omega-3 fatty acids?
This is also a very interesting question. I believe, or let me say, it's not only belief. I recommend to start with the intake of omega-3 as early as you can. Along with the guidelines, national and international guidelines, I recommend to start with the lipid emulsions as early as we can. There's no reason to wait. You know, we have older recommendations, very old recommendations. They told us, okay, you can take six or 7 days and then start with lipid emulsions. This is not longer valid. This is obsolete. Today, we recommend to start if the patient really needs artificial nutrition, if the patient needs parenteral nutrition, then we should start with the lipids as early as we can on day 1. If the patient is in a hemodynamic, stable situation, we should start with parenteral nutrition, also with enteral nutrition, of course. And when we do this, then we should start with omega-3. Why? Because I tried to show in my presentation the problem is the inflammatory process just in the beginning after a trauma or a huge OR. And this inflammatory response is coming very quickly. If we wait with the intake of lipids, if we wait with the intake of omega-3 2 or 3 days, we are too late. We are definitely too late to make this resolution of inflammation. We have seen this from different papers published in international journals. If you come too late, and if you use a bolus application after 2 or 3 days, more or less nothing will happen. You cannot get the benefit of this idea of resolution of inflammation if you wait too long. So again, lipids are an integral part of every type of nutrition regimen, independent of if enteral or parenteral. We discuss here in this situation the parenteral nutrition. If the patient needs parenteral nutrition, then we should start as early as we can with lipid emulsions in combination with carbohydrates, amino acids, of course. And please go with omega-3 at once.
All right, good. So we can actually summarize that you recommend it as early as possible and basically as a standard therapy.
Definitely.
Now, there are two other questions which I actually can combine with regards to the daily requirements and dosage of omega-3. What do you think would be the most optimal dosage of omega-3 fatty acid, and is there a maximum dose for EPA and DHA?
Okay, I showed you in my lecture the recommendations from the European guidelines, and this is very close to the German guidelines, of course. First, we should follow the idea, what is the maximum intake of lipids when we go with parenteral nutrition. We should not exceed 1.5 gram per kg body weight per day of lipids. And please keep in mind, you should also calculate the amount of the non-nutritional lipid sources, like if you go with propofol.
For sure.
With regard to a critical ill situation in an ICU ward, most of the patients are on a ventilator and they need some type of sedation, of course, and it's very popular to do this with propofol. This is also a lipid emulsion. Please keep in mind, this must also be calculated, but the maximum load of lipids should be 1.5 gram per kg body weight per day. And we recommend the dosage of EPA and DHA should be in between 0.1 and 0.2 gram per kg body weight per day. And this is, from my point, absolutely a safe dosage window. We should not overwhelm this. We should not go with a higher dosage from my point of view. So if you follow the international guidelines, and they are well accepted, we have this maximum of 1.5 and a recommendation of EPA and DHA of 0.1 to 0.2 gram per kg body weight per day. I believe this is perfect, and yet you are on the safe side, and you can get the benefits of resolution of inflammation with this type of dosage.
Absolutely clear. So clear statement on that. And we have another question. It's with regards to the European pharmacopoeia monographs you've mentioned. So could you please highlight again the difference and also the importance between, respectively, the differences between these two monographs? So one of the questions here.
Nadine, perfect. Good question. We have definitely two different European pharmacopoeias available. In the past, I tried to show the results of David Driscoll. He made some years ago this type of investigation, and he could show the difference of two different commercially available products. It was the b.braun product and the competitor. And he was also able to show many years ago that the EPA and DHA concentration is definitely higher in the lipoplast lipidem product. Okay, you mentioned the European pharmacopoeias. The pharmacopoeia 1352, it's the one for the lipoplast lipidem product, is telling us EPA and DHA content of minimum 45%, and the total omega-3 acid content should be 60%. And lipoplast lipidem is exactly in this window of concentration. The second European pharmacopoeia, 1912, is definitely lower. They have an EPA and DHA content of maximum 22%, and the minimum total omega-3 fatty acids of about 28, maximum 30%. So you can see from this difference in the concentration that the competitors, and I can tell you, all of the competitors are in this dosage or concentration is definitely lower, is significant lower compared to the b.braun product. So if we follow the idea of using omega-3 in our parenteral nutrition, I believe it's the best way to go with the b.braun product, because you can get the highest levels of EPA and DHA, but you can keep in the dosage window, as we just discussed a few seconds ago. The differences between the products is coming from different production procedures. I believe it's not necessary to declare this in detail, but this is the story behind. But again, the content of EPA and DHA in lipoplastin lipidem is perfect.
Thank you so much for highlighting that once again. Well, so looking at the time and the chat, I believe so far we don't have any more questions. So thank you so much, Dr. Adolf, for joining us today and for this interesting questions and answer session. We see you later probably. And so we will proceed right away with our next speaker. Oh, we have another question. I would like to take that one. Just another one I got in, so we can take 1 more minute. How many grams lipid per kilogram body weight from the b.braun omega-3 source will be needed to provide the 0.1, 0.2 grams of EPA DHA you have just mentioned per day?
Okay, this is a matter of calculation, of course. I believe if you follow the recommendation I just gave you, a maximum of 1.5, including the non-nutritional lipid sources. If you go with this and if you calculate also the propofol intake, you are about the level of 0.1 gram per kg body weight per day, a little bit lower, but you are actually in the recommendation, of course.
All right. Thank you so much. All right, then. Once again, thank you so much. It was a really true pleasure having you here, Dr. Adolf, and thank you for your great contribution. And we will proceeding right away with our next lecturer, Dr. Norbert Nagel, with a closely related topic, namely guideline recommendations in nutrition on the ICU. And not to keep us waiting any longer, I'm directly handing over to you, Norbert. All eyes are on you.
Thank you very much for the kind introduction, and I also like to welcome the audience, and I'm very happy to be with you this afternoon and this evening. So within the next few minutes, I will go with you a little bit through the guideline recommendations for the nutrition in the ICU, connected with the question when to do it, what to do it, how much to give it, and how to perform it. So here are my disclosures. And we will shortly, let's talk about the ICU patient. Then I will go into details with the recommendations in the ICU. We will see how to do it. And finally, end this lecture with a take-home message. So what is the ICU patient? You've already seen this picture in the presentation of Professor Michael. So I will not go into details here, just only to remind you that the critically ill patient is a very complex patient. And it's characterized by a number of organ dysfunctions, and you all know certain scores like the SOFA score or the Apache 2 score to characterize the severeness of his illness. So I do not want to go into detail here, but go on with the phases of the critical illness. You also have seen this picture before. However, I will lay my stress a little bit on some other things related to nutrition. We've seen that we have several phases. The acute phase was an early stage, was a late stage. Then, depending on the amount of homeostasis incident here, the patient either ends up in a post-acute phase in reconvalescence and recovers and goes to rehabilitation, or when it's severe illness and the patient has a very severe incident, then he can go into a chronic phase and to a reconvalescence phase. Normally, these phases have a certain duration here. The early phase is that it's about 1 to 3 days after admission to the ICU. The late phase starts 2 to 4 days later, so that when the patient gets through this acute phase, after about 7 days or more, he ends up in the post-acute phase. Well, with regard to nutrition, and we come to this in a little bit in detail a little bit later, is that the early phase of the acute phase is characterized by a massive amount of catabolism. And you cannot overcome this catabolism. Why, I will explain a little bit later. In the late phase, you have more or less a mixture, and you go from a catabolic to an anabolic state of this patient. And when the patient gets into the reconvalescence or into rehabilitation phase, then he is in totally anabolic metabolism, and now you can really build up muscles again. However, when the patient ends up in a chronic phase, he's also in a very catabolic stage. And this has to be taken into consideration when considering parenteral or enteral nutrition in these different phases of the critical illness. So now what are indeed the recommendations from the various scientific societies? So let's start with the question, when to start, generally when to start nutrition at all. And here is the recommendation, and the general recommendation, really to start with enteral nutrition. Enteral nutrition is always first. However, the guidelines differ a little bit in the timing when to start it. So the DGIM and the ASPEN says, yes, it should be started within 24 hours. The ASPEN, the European guideline, says it should be started within 48 hours. But in any case, you should start very early with the nutrition, with enteral nutrition. And only in case enteral nutrition is not possible or contraindicated, you would go either to a total parenteral nutrition, or when EN is only insufficient, you go to supplemental parenteral nutrition. However, there's a big difference between the European view and the American view when it comes to parenteral nutrition. So when there's a contraindication to oral or enteral nutrition, European guidelines says you should start PN after 3 to 7 days. Well, in the US guidelines, they say that exclusive PN should be withheld over the first 7 days in patients with low nutritional risk. So no parenteral nutrition in the first 7 days when there is no risk. However, they should initiate exclusive parenteral nutrition as soon as we have a patient with a high nutritional risk, may be characterized by a nutritional risk screening score of bigger than 5, or a Nutrix score also bigger than 5, or in a very, very severely malnourished patient when enteral nutrition is not feasible. Supplemental parenteral nutrition can always be considered after 7 to day 10 days, when the patient cannot meet his nutritional requirements via the enteral route. So why is it like this? So here you see a conceptual illustration of the energy metabolism. When you remember in the history of parenteral nutrition, it was very soon clear that critically ill patients in the ICU have massive muscle mass loss. And this in the first time was tried to overcome by the first concepts by Stanley Dudrick, hyperalimentation. So the patient was fed with very, very high amount of calories, 50 kilocalories, 40 kilocalories per kilogram body weight. Just give him very, very much of calories. This was found to be not beneficial for the patient over the years. And the main thing here is mechanism called autophagy. I do not want to go into the details here, but autophagy is a process to for the body to get rid of the garbage after trauma or something like that. But this does not work when the patient is overfed. And you can see it, or it might become clear when you really see that in the development of mankind, there was no ICU. So when the people get sick, or when the people were injured, there was no ICU. So the body has to develop mechanisms to get over this. So in the first phase here, in the early period of the acute phase, the energy comes from endogenous sources, and you cannot overcome this with exogenous energy production. However, after a few days, the body starts to get more and more used to also to get or to use energy from exogenous energy sources. So when you see here, this line here represented by A is the endogenous energy production of the patient. Normally you should measure it with indirect calorimetry. However, with indirect calorimetry, you can only measure the total amount. You cannot measure the amount, how much energy really can be consumed by the patient. So the recommendation in generally is, and we will see that later also, that you start very, very slowly and ramping up the calories, not to overfeed the patient. Underfeeding or hypercaloric feeding is not so bad than overfeeding to the patient. And the optimal energy is that you add so much energy to this patient and give him so much energy that you in the end up with more or less an energy provision, at least at the line A you can see here. So the thing is, you should do a stepwise increase of the energy supply in these patients over the first days in the ICU. So how much should be given? As I told you before, the first concepts were with an hypercaloric concept. Now you're more on the other hand side. And the recommendation is, if you cannot use indirect calorimetry, and here are more or less the recommendations are equal with between all the scientific societies, it's around the maximum of 25 kilocalories per day when you use predictive equations. Otherwise, the gold standard is still indirect calorimetry. However, not everybody has such indirect calorimetry. So it's really recommended to start slowly and to go more for a hypocaloric feeding in these patients in the first days. So now, what about the protein? And here it is very, very important that you have the right balance. So, as I said before, it's not good for the patient because you block autophagy when you give too much energy in the first days, but it's the same for protein. So in the first days, and all these guidelines here more or less are the same, say that you also should, with the energy, you should also ramp up slowly the protein intake. The German guidelines here said that it should be around 1 grams of protein should be given. The Aspen guidelines as well, 1.3 grams of protein equivalents. And the Aspen guideline and from the US recommends a hypocaloric parenteral nutrition with at least 1.2 grams of protein. In the later phases, when the patient gets more from a catabolic to an anabolic state, also the amount of protein should be increased. There is not so much evidence, but they say around at least 1.5 grams of protein. And the US guidelines even go much higher to 2 grams of proteins. This does not include the proteins which might be used, or the additional proteins that might be used when the patient is on a continuous renal replacement therapy. In this respect, of course, the protein losses into the dialysis has to be compensated by additional amounts of protein. This has to be taken into consideration. The European guideline makes no special recommendations for the late phase and for the chronic phase. They say that we should provide 1.3 grams of protein equivalents, and it should be given progressively here. So you have also to take into consideration that you give energy and protein together. They can mostly not given separately. And there is a nice retrospective court study by Sussman, and they found that in this picture you see the unadjusted ratio for the 60 days mortality as a function of the provided energy, here the calories in percent of the resting energy percentage, and they found that there is a optimum of around 70% of the measured resting energy expenditure. And therefore, the Aspen defines an isocaloric feeding of the defined target when you are between 70 and 100% of the measured target. Because you see here, you have more or less an U-shaped curve. So a too high underfeeding is not good, but also overfeeding is also not beneficial for the patient. On the other hand side, when you look to the protein, it seems to be that the more the better it is. However, the question is this really the case? And therefore, I show you this nice study and nice paper published by George Kreiman and his group. And he investigated the energy nitrogen ratio as function of the total protein loss. So this is a parameter which is very important to characterize a nutritional regime because it shows how much non-protein energy is available really to use the protein in the body for muscle synthesis. And when you see when you have a very, very high amount of energy that can process the provided protein, you see there's also a very low amount of protein loss. However, when this ratio changes and you have too less energy and too much protein, you have a higher protein loss. So we've seen before you have hypercaloric feeding and you have a certain amount of protein to give. So you have to find the right balance here. However, you should also take into consideration that the additional protein loss you have here in an ICU patient might be due to an increased protein loss because you cannot use the protein here and you have loss of unutilized nitrogen because you have not enough energy. So the balance here is extremely important. So we have seen we have energy, we have the protein, but we also have to give, of course, non-protein energy to the patient. And the energy of choice in parenteral nutrition from carbohydrates is provided by glucose. There is no recommendation for the glucose from the US Society, and some recommendations from the German and from the European Society, which at first glance look a little bit different. So the recommendation from the Aspen, which is very simple, they say it's safe to give about 150 grams per day of glucose. The old recommendation said you have to give at least 2 grams of glucose. In Germany is to say, yeah, it should be below 4 grams of glucose. And in case you have a reduced glucose tolerance, like in diabetes, you should give only 1 to 2 grams of glucose. So what does this mean? But when you calculate it, this 150 grams per day of glucose really corresponds to 1 to 4 grams for a body weight range from 37.5 to 150 kilos. So indeed, the recommendations for glucose does not really differ so much between these scientific societies. Now, what about the lipids? Professor Michael already told us about this, so I do not go into the details here. I just want to point out that you have to have the right balance between the non-protein energy from lipids and the non-protein energy coming from glucose. And here is the recommendation to be careful. Too much glucose can produce hyperglycemia, which must be treated with insulin. However, on the other hand side, when you give too much lipids, you can induce hypertriglyceridemia. So you always have to also to find a balance between the amount of glucose and the amount of lipids. And you also always have to take into consideration that 1 gram of glucose provides 4 kilocalories and 1 gram of lipid provides about 9 kilocalories. However, as already pointed out before, there are some differences between the amount of fat which should be given. In Europe and in Germany, fat is an integral part of the parenteral nutrition. However, in the US, they limit the amount of at least here soybean-based intravenous lipid emulsions to a maximum of 100 grams per week, maybe given in 2 doses. So here are some differences. As said before, the non-nutritional energy sources must be taken into consideration. And when you think about a patient on the ICU, which is maybe sedated by propofol, propofol is prepared in a lipid emulsion, and a 10% lipid emulsion with 1 gram, 0.1 gram lipids per ml provide also 0.9 kilocalories per ml. So when you give this patient 20 mLs per hour of propofol, this corresponds to 430 kilocalories per day. So this has to be taken into consideration when you make the balance for your nutritional regime. Also, when you give citrate, citrate can also amount between 150 and 280 kilocalories per day. So this also has to be taken into consideration here. So we have already seen this, therefore I will not go into the details here. Professor Adolf has pointed it out that there are different recommendations. However, it's a clear trend for omega-6 reduced fat emulsions and for omega-3 enriched fat emulsions. Here I show you a very recently published meta-analysis, which was not shown before. It's from the group of Christian Stoppe and Daryl Hayland from Canada. And they analyzed only ICU patients. And they only took studies which were done in the ICU, and they had a very, very tough selection here. And they found that in generally, when you reduce the omega-6 fatty acids in the parenteral nutrition, like in mixed fat emulsion, MCT, LCT, you have a reduction in the hospital length of stay. However, there is no effect on mortality. When they investigated the subgroup here with the fish oil containing parenteral, they found that the hospital length of stay is further reduced, so this stays. And as a result, you have to decrease in the risk of infections. This was already shown before. So you can sum up here at that point that really omega-6 reduced and fish oil or N3 fatty acids enriched fat emulsions have a favorable impact on these patients, especially with regard to the hospital length of stay. And the fish oil may have the greatest clinical impact. So the future of the lipid emulsions, I think, will be an omega-3 fatty acid enriched fat emulsion. So now you've seen all these recommendations, and now we have to bring more or less everything together. With regard to the energy requirements, indirect calorimetry is recommended. However, when you do not have such a machine, you can use the ventilator. There are also formulas to calculate the energy expenditure from the ventilator, or you can use the predictive equations. Then we have seen that we have different phases. And in the first acute phase, hypocaloric feeding is recommended, starting with less than 70% of the target in the early phase, and then slowly ramping up to about 80 and 100% below day 3. So you have a slow ramp up just from the beginning when you give the nutrition until you end up to the target after the end of the acute phase. Then when the patient ends up in the chronic phase or in the recovery phase, you have to provide higher energies. The reason behind that is that we have seen before that in the acute phase, you have more or less a catabolic state, and the energy and the protein you give is mainly not to increase the muscle mass or to build up muscle mass, but to reduce the muscle loss, because you cannot really build up muscles in these phases. However, there are some methods that when the patient is cycling or when you have some some impulses for muscle for building up the muscles, you can do that, of course. However, in the chronic and especially in the recovery phase, we need more, or the patient needs more energy really to process and to metabolize the proteins to really be in an anabolic state and to build up the muscles from the provided protein. And this is pointed out here with an increase to maybe from 25 to 30 kilocalories or even to 35 kilocalories per day. On the other hand side, you have the protein requirements. It was the same, that should be given progressively. So you ramp up also the protein over a few days. So to end, as it's not very slowly, then you end up with the 1.3 grams of protein equivalents. And in the chronic and the recovery phase, you even need more. So you increase also the protein amount here. However, this has an impact on the design of the nutrition you have to give in these phases. So maybe when you start very slowly with 25%, 50%, 75% over the first days in the acute phase, then the nutritional regime is, when you calculate it, has a nitrogen calorie ratio of about 1 to 60, 1 to 80. When you go to the chronic and the recovery phase, when you increase the energy and the same amount increase the nitrogen. However, you end up with ideal regimes according to the guidelines with a nitrogen calorie ratio ranging maybe about from 1 to 100 to 1 to 120. So now the question is how to do this. This first looks very complicated. However, there are, of course, solutions for that. So you can either use commercially available ready-to-use 3-chamber bags. They provide amino acids, glucose, and lipids within one bag, ready to mix. So just by pressing on the chambers here, you open the peel seams and you have a ready-to-use nutritional regime. So it's very easy to use. There's different volumes, of course, available that you can adapt the nutrients to the needs of the patient. They can be stored at room temperature and they have a shelf life of 24 months, which is very convenient for you. So they are easy and fast to use. And of course, it's important that the 3-chamber bag not only provides the macronutrients, but for a complete regime and to avoid any deficiencies, you also have to add maybe additional electrolytes, but you have to add vitamins and trace elements to these regimes. And therefore, also compatibility data are available here that you can do this safely. On the other hand side, you have to see that here the nutrient relations are fixed, and that the guideline compliant nutrition might not be possible for all patients and for all body weights. So you have to see, when you calculate a regime, let's say for a 70 kilo patient, everything fits, that when the patient is lighter, you give more calories, more protein. And when the patient is heavier, you give less calories and less protein. So you have to take this into consideration here. On the other hand side, of course, you always have the possibility of doing a compounding. When you do the compounding, it's very easy. You can absolutely adapt the nutrients to the patient needs. You also can really make a measure with indirect calorimetry and can tailor the regime. You can do this by patient, of course. Then you have to calculate for each patient the regime. So you have to have somebody in the ICU which is really used to do this and who has the knowledge to do that. Or you can do standardized compounding regimes where your hospital pharmacy produces standardized compounded regimes which can be stored and you can use them like a 3-chamber bag. For our b.braun products, of course, we also have compatibility data and stability matrices for compounding bags. However, you have to take into consideration that these bags have a limited stability, maybe up to 30 days. However, in special cases, maybe they're only stable for 7 days. This just depends on the nutrient relations and what you additionally add to these bags. What is absolutely mandatory in these cases is that these bags will be stored in the fridge. They have to be cooled, also cooled. You also have a cool transport and a cool storage. Otherwise, these bags cannot be used. So cool storage and cool transport here is absolutely mandatory. And of course, you have to have the special equipment for the compounding. So now let's sum up. We have seen that the nutrition in the various phases are a little bit different. So just depending on the nutritional status of the patient, you can say that when you have a malnourished patient, that you should not feed him in the very early phase of the acute phase. You can go to a total or supplemental nutrition when needed in the late phase, and it's the same for the recovery phase, that these patients can have a total or a supplemental nutrition just as they need it. When you have a severely malnourished patient or when there's the risk of the refeeding, you should be careful and start with supplemental nutrition with very conservative nutritional goals, hypocaloric feeding we've seen here. And in the later phases, it's always the same, just as the patient needed, either total or supplemental parenteral or enteral nutrition. So, what take home from this lecture? We have seen that we have a phase-adapted nutrition with different nutritional requirements in the very early acute phases, in the late acute phase, in the chronic and the rehabilitation phase. In generally, energy and protein should be ramped up over 2 to 4 days to the targets. You should use indirect calorimetry for the determination of the energy expenditure, and only when this is not possible, then you can use the ventilate data from the ventilator, and if this is not possible, then you can use predictive equations, and we have seen it's around 20 to 25 kilocalories here. The hypercaloric feeding here is recommended when you use the predictive equations, so you can go just to the low side of this range, which I told you before. I also have seen and discussed for several times today that you should use omega-6 fatty acid reduced fat emulsions, but to really get the best clinical benefit, you should go to the third generation lipid emulsions like Lipoplus and use N3 fatty acid enriched lipid emulsions. Of course, you can do this also with ready-to-use 3-chamber bags as convenient solutions, or with the compounding to really tailor the nutrition to the patient needs. So when you do everything like this, you are really at the top, like on this nice building. And I say thank you very much for your attention and for your time. And now we are open for the discussion.
Thank you, Norbert, for this insightful presentation, and welcome back to another question and answer session within the framework of recommended nutrition on the ICU, and now specifically focusing on nutritional guideline recommendations. Let us start with the following. Norbert, you have mentioned the latest guidelines and recommendations for the use of lipid emulsions in TPN, as well as difference in the usage of lipid emulsions in different areas around the globe. Now, why do you think there is such a big difference?
Yeah, I can only guess, but I know that there was in the late 70s, there were lipid emulsion, especially in the US. And this was a very bad lipid emulsion. It was Lipomul. It's not on the market anymore, and they had very bad experience with that lipid emulsion. It was long before the soybean fat emulsions came on the market. And so I think this might be an explanation for the reluctance of giving too much fat in the US, especially in the US. But when you look in the current literature, with now that new and third generation lipid emulsions are also available in the US, this might change because they are now very happy to have this fat emulsions, and all the reports and all the papers published here, they are very, very positive, and they see the benefits of this lipid emulsions, of these new lipid emulsions, and so maybe there will be a change. This is my personal opinion, more or less based on a little bit in the theory of lipid emulsions and what can happen. This is also a little bit based on the work of David Driscoll, who worked very much on the quality of lipid emulsions and on the specifications, and that really the lipid emulsions get more safer for the patients.
Thank you for that. And I think that goes along with the next question. If you could add on a little bit more on the safety of the application, of today's available lipid emulsions. You have mentioned that already, and you've just highlighted that again, but maybe once again, let's say as a summary.
Yep. Lipid emulsions are not only to provide energy, they're also to provide essential fatty acids. That's why it's absolutely essential to give them. However, lipid emulsions also have some inflammatory properties or anti-inflammatory properties. They also have immune properties, and that's the thing which you have to take into consideration. Especially the pure soybean-based fat emulsions are very, very rich in linoleic acid and omega-6 fatty acid. And when this omega-6 fatty acid is incorporated into the membranes, it drives the cell more and more in, let's say, a pro-inflammatory state. So this was recognized pretty soon, and then the second generation of lipid emulsions came into place, and they added MCT, medium chain triglycerides, which are more or less immunoneutral, but they only reduce the absolute amount of linoleic acid, but the balance between omega-3 and omega-6 were more or less the same. So the third generation just said, okay, we have to add more omega-3 fatty acids to it to get a better balance of it. And we know that all the Western diets are very low in omega-3 fatty acids. And as we have seen before, omega-3 fatty acids have very, very important inflammatory and immuno properties. And therefore, the whole system, inflammatory system, when you add additional omega-3 fatty acids, is shifted into an anti-inflammatory state. And this is very important. Therefore, it is really important to have a good balance. And today we know that the balance should be around one part omega-3, 2.5 parts of omega-6. And this is exactly the ratio we have also in our LipoPlus.
All right. So basically you're saying what's available today is actually safe to use. And that goes along with the comment which we have received via the chat. It's not a question, it's a comment. I think that the Aspen guidelines, or the American guidelines, limit lipid to 100 grams per week, as at the time, by that time when they were published, only 100% soy lipid was available and not the mixed lipid. So I think that goes a little bit along with what you've just mentioned, right?
Exactly. Yeah. So now the new lipid emulsions also came available in the US with the reduced amount of omega-6, with a higher amount of omega-3, with now a much better quality of the emulsion itself. So all this goes into the direction of a more safe lipid emulsion. And the next thing is, and I think Michael already pointed it out before, is really, especially in the ICU, to limit the amount of lipids to 1.5 grams. And also including the non-nutritional sources here, not to overload the patients with lipids. It's a different situation when the patient is on a dialysis, either in an acute dialysis situation or on chronic dialysis. Then you need much more, because these patients lose energy, and then they maybe also need more lipids in that case.
We've actually heard during the past two presentations of Dr. Adolf and yourself about the advantages of omega-3. You've just mentioned the immune modulating effect, but could you just summarize a little bit the pharmacoeconomic advantages of the supplementation of omega-3 fatty acids in fat emulsions?
Yeah, we have also seen some results of meta-analysis where more and more gets clear that this lipid emulsion has also economic benefits. And this is mainly based on the evidence we now have that omega-3 fatty acids are really able to reduce infections and nosocomial infections, especially in ICU patients. One example is the ICU lipid study here, which showed this very clearly. On the other hand side, also, it's now very clear that these lipid emulsions can reduce the length of stay in the hospital. So when you put these two things together, treating nosocomial infections is very expensive. And also when the patient does not stay too long in the hospital, it just became clear that this must be some savings. And this is also the results you have from the meta-analysis here and from the economic evaluations you have now. So you see that now the evidence we have reduced inflammation, shorten the length of stay, also results in an economic benefit when these emulsions are used.
All right. I would like to take the last question. Speaking of evidence, some guideline recommendations are actually based on weak evidence. However, our knowledge about metabolism of critically ill patients is constantly increasing. So how likely do you think is that these recommendations will change in the future?
Yeah, when we look into the past, going back to the days of Stanley Dudrick's revolutionary work, so the first thing was to give much calories. Hypercaloric feeding at that time was the strategy. And now we know that this is not beneficial for the patient. We got a little bit deeper into the metabolisms and now also found why is it like this. There is the process of autophagy, and we know now that this process is more or less limited and inhibited when we get too much proteins, too much energy right in the first phases of the ICU situation and in acute phase. So we now see that the whole system is changing and that we get more insights into the post-aggression metabolism here. Therefore, I think that the guidelines will not change now in very big ways. So maybe there will be some slight adjustments on what we think is the best balance between calories and protein. But I think it will not change pretty much because the human metabolism stays the same for, let's say, since the Stone Ages. Yeah, and at the Stone Ages we did not have any ICUs. So when the patient or when some hunters got hurt, they had to see for themselves how they can heal. And this is the stage of our metabolism still today. We can support them with the ICU, but we cannot change it. Therefore, when we get put in too much protein, too much energy in the first days, they are useless. And then they are also not beneficial. So I think there might be some slight changes with regard to the absolute amount of energy, of protein, which should be given, but not basic changes that the whole picture will turn.
Thank you so much, Norbert. That was a very interesting final statement from your side. Time is actually up. Thank you so much for your great presentation and this very lively, active question answer session with you. Great to have you on board with the NutriTalks. And it's now 12 o'clock CT, or actually 12:03 to be honest with you. And at least in Germany, that equals time to eat. And to be fair with you, after all this brain food, I'm in the mood for having real food. So I propose for us to step into a short lunch, dinner, or snack break, depending on your location and the respective time zone you're dialing in from. Let's recharge our batteries and let's meet back again in 1 hour, respective 1 p.m. CT. Please also be encouraged to have a look around our booth and information material, which I've mentioned in the beginning, throughout your break. And as we say in German, guten Appetit and see you at 1. Dear ladies and gentlemen, welcome back to the third subject area of our NutriTalks training event. I hope you had an enjoyable break and are curiously looking forward to next speeches to come. The next block will be dealing with home artificial nutrition, an important and growing sector which we are welcoming two speakers for. Dr. Ioni de Brito-Ashurst is the Head of Therapies and Rehabilitation at the Royal Marsden NHS Foundation Trust. Most of her clinical work has been critical care and chronic illness and the treatment of malnutrition, which has led her to active research interest in the prevention and management of muscle mass. She will kick off the first part by sharing a 21st century perspective on home artificial nutrition. The second session will be presented by Dr. Riccardo Caccialanza, Director of the Clinical Nutrition and Dietetics Units of the Research Hospital Fondazione IRCCS Policlinico San Matteo in Italy, and contract professor at the University of Pavia. He is specialized in clinical nutrition and has expressed his profound knowledge in clinical nutrition in more than 100 publications. He will show us the importance of vitamin K in long-term home parental nutrition. But let's get started with home artificial nutrition at 21st century perspective, presented by Dr. Brito-Ashurst. Ioni, all eyes are on you.
Thank you so much for the invitation, b.braun, to talk about home artificial nutrition support, something that I feel really passionate about it. In my talk, I will be covering modes of nutrition support at home, which is enteral nutrition and parenteral nutrition. We'll be looking at the key considerations when planning these modes of nutrition, guidelines, what are the indications, what group of patients are suitable for this type of nutrition, and then thinking of patient considerations, patient lifestyle, how do we plan this nutrition around the patient, and most importantly, consider monitoring, how we're going to monitor the efficacy of the nutrition therapy. So let's start talking about home enteral nutrition. We will cover indications, suitability of the patients. Those are international recommendations that we'll be presenting. Looking at how we can deliver that nutrition, nasogastric, gastrostomy tube, the most common form of nutrition in the community, jejunostomy, and then formula, what teaching the patients need, and the discharge process. These are recommendations from the ESPN guideline on home enteral nutrition. And essentially, what ESPN is telling us is that for all those patients at nutritional risk or malnourished who cannot meet their nutrient requirements by normal dietary intake, but obviously have a functioning gastrointestinal tract, that nutrition support, home enteral nutrition is recommended for those patients. And if we are feeding those patients at long term, and that means over 5-6 weeks, then the preferred mode is gastrostomy, or for those that have malabsorption, jejunostomy. Okay, how are we going to provide that nutrition? So access, it could be nasogastric feeding. If we are talking about the short-term nutrition within 1 month, 5 to 6 weeks the most, we need to think about the comfort to the patient. You know, that tube in the nose can have erosion of the nose bridge. I'm going to start that again. Let's think about the access, how we can provide nutrition support. Nasogastric, if it's a short-term feeding, we need to think about comfort to the patient, or if it's long-term feed over 5-6 weeks, then we need to look at gastrostomy, and that could be surgically placed or endoscopically placed. Most type is a PEG. Or for those patients that malabsorption is a problem, pancreatitis, then we need to consider jejunostomy feeding. So we've decided we're going to feed the patient gastrostomy. We've placed gastrostomy on the patient. When can we start feeding that patient? And historically, it would take about 24 hours before we can actually start feeding the patient. The first 12 hours would be observing for the symptoms, monitoring the patient, the usual blood pressure, temperature control, and then we would spend the next 12 hours giving water through the tube to just check for potential leakage. But now recommendations are that we really don't need that long. Within 2 to 4 hours, we can start feeding the patient. So you spend a couple of hours checking, doing those nursing monitoring. And then after that, we need to check to see if there is leakage. So you could either use a pump to deliver some water or use a syringe and flush some water to the peg and observe for some potential signs of leakage. If it's fine, then we can start feeding. So reduce that waiting time considerably. Start feeding within 4 hours. So we're going to start feed. At what rate are we going to give that feed to the patient? And it very much depends on the patient nutritional status. So if your patient was eating before, even if it was a small amount, you can start at 50 meals per hour. And then you increase gradually at 20 to 30 meals at a time every 4 to 6 hours. Or if you want to be a bit cautious, your patient is malnourished, then let's start at 30 meals per hour. Again, increase gradually every 4 to 6 hours by 20 or 30 meals. So you've started at 30 meal. After 4 hours, you go to 60, and then you go to 90. So within 12 hours, you might be reaching the patient goal. What about those in jejunostomy, jejunal feeding? We have to be a bit careful about the volume we give into the patient. The jejunum does not have the same capacity that the stomach have. So we start slowly. And Hans and Gertz showed here that they started feeding with your standard polymeric feed, 1 calorie per meal. They started at 30 meals per hour, and it took them up to 3 days to reach that desired a target. So with the jejunostomy, you want to take a bit longer to progress the feed. So you might wait 8 or 12 hours. So we start at 30, then at 8 hours or 12, you go to 60, then another 12 hours, you go to 90. So that could take a couple of days to reach the target. Okay, how we're going to deliver the feed, and we can do that with the most used method, which is pump. That's expensive. You need electricity. The patient is tied to the pump, although nowadays the pump are really small and can be, fit in the backpack. But you need education of how to use that pump. And then one of the benefits is you know how much we're receiving, the exact delivery, and also reduced risk of gastrointestinal intolerance and aspiration. And you can deliver it cyclic for a short period of time or continuous for a longer period of time. Or you can look at inexpensive methods like gravity way of delivering, and it could be a gravity bag. You don't need electricity. You just need a bedside pole or hook to hang that gravity. It's easy to use, it's portable. The problem is you don't really know, the delivery is not as exact as with the pump, and is given over a small amount of times, within 1 hour or so. And then you have bolus feeding with a syringe. So you would have a 50 ml syringe with a plunger. You would put the feed through and just flush that feed through the peg. So it's inexpensive, no electricity is needed, not tied to the pump, very easy to use, portable. Again, delivery is not as exact as with the pump. It is a fast way of delivering the feed. You can deliver within 1 to 2 hours, the bolus, but there is increased risk for vomiting and aspiration. And you really need to make sure patient head of bed or is elevated, or ideally patient is sitting down. So how are you going to deliver bolus feed? So let's think of a case scenario. We have a patient that he needs, you calculated nutritional requirements, and he needs 1600 of calories a day. And you're going to start it with a standard feed of 1 calorie per meal. So he needs 1600 meals of feed a day. You divide that feed into 4 to 6 bolus. So let's say we divide it into 4 bolus of 400 ml. So the patient could have 4 bolus of 400 ml. Infusion volume on the bolus can be between 200 to 400 mils. And how would you start that? Gradually, you can use a syringe, 50 ml syringe with a plunger, put 50 mils in, flush that very slowly, wait 15, 20 minutes, then you can flush another 20 mils, another 50 mils. 2 hours later, you flushed again, and just observe, is he okay with 100 mils? Then let's move to 150. Then let's move to 200. And gradually you will reach that tolerance of the patient, but it's usually between 200 to 400 mils, to tolerate that bolus. And there is no evidence that bolus feeding predisposes to diarrhea, bloating, aspiration, and some GI intolerance. It's a more physiological way of feeding as well, because you could feed, following the eating pattern at breakfast and lunch and dinner. And I really like this way of feeding, because if we think about the patient with chronic illness, the older adults that have anabolic resistance, and the studies have shown that the high protein bolus, between 20 to 30 grams of protein, feeding at a time, that dose will break down that anabolic resistance. So this is a good way of providing a high protein bolus to the patient. Then there is continuous intermittent feeding that we need the pump to deliver. That is a better way for jejunal feeding. The jejunum does not have the same capacity as the stomach. And the feeding pumps, when I started nutrition over 20 years ago, they were very big, but now they're very small and can be, fit into a rucksack. Or you can do a combination of methods really to suit your patient. So if we think back of our patient that needs 1600 calories and we're going to give 1600 meals of feed, we could feed him 100 meals per hour over 10 hours overnight. So let's say for 9 at night until 7 in the morning. So he gets 1000 meals, 1000 calories during the day. There is 600 calories to be delivered. We can use 2 oral nutritional supplements, put in a syringe, and slowly flush through the gastrostomy. And that could be given at lunchtime and dinner time. So a really good combination that it gives time to the patient. During the day, he's not attached to the pump, but during the night, he's receiving everything else that he needs. Formula selection, there is a vast range of formulas out there. Most commonly used are polymeric formulas. And, in terms of calories, it starts at 1, goes up to 2 calories per kilo. And you can, calories per meal, and you can have feeds with fiber or without fiber. Obviously, fiber supports that regular bowel motion. It has prebiotics as well, the fruit oligosaccharides. And you can have the standard feed or those concentrated high protein feeds. Or you can have peptide, the hydrolyzed feeds where protein are presented as peptides or amino acids, or a specialized formula like diabetic, renal, or pulmonary failure formula. Okay, so you're going to discharge the patient home. You need to identify what is the best regimen for the patient. Consider lifestyle. If it's somebody active, maybe they can be bolus feeding, or maybe it's just intermittent cyclic feeding during the night. So during the day, they are free to do whatever they want. Identify the best formula for the patient. What about fiber intake? How much fiber does the patient need? You need to work out with your patient how much fiber do they normally have in their diet. In the Western diet, is usually between 18 to 23 grams of fiber. So identify with the patient how much fiber he is. If he's not sure, then go for between 15 to 20 grams of fiber. In my experience, patients will do well in that amount of fiber. If we do too much fiber, the patient can have a constipation. And then we need to think about water flushes as well, hydration for that patient. So you know that you need to flush between 30 to 60 mils of water before and after medication. And then you need to calculate patient's fluids requirements. Recommendations are about 30 to 35 mils per kilo. You see how much fluid the feed is providing, how much you give in after medication, and then what is left, you can either deliver through the pump or with flushes with a 50 ml syringe. You need to identify who is going to write that nutrition support prescription, the training that the patient gives. And obviously, if he's using the pump, then he will need a bit more training, and give all the contacts to the patient, the community, the company that is providing that, those community professionals, dietitian, nurses, and obviously details for the supplier and details at the team in the hospital that follow the patient. Monitoring, really important to see if our way of feeding the patient is effective. So we need to consider weight. What is the patient BMI? Is there weight loss? Look at percentage of weight loss, and cannot neglect muscle. Muscle is extremely important and is associated with morbidity and mortality, low muscle mass. So we need to look at muscle mass and muscle strength, hand grip, bioelectric impedance assessment, anthropometric measurements, and some functional parameters. And I will be talking about some functional parameters later on. Looking at hydration status, is the patient having enough fluid? Looking at the tolerance, nausea, vomiting, diarrhea, and biochemistry to really check if the patient is getting everything, how well they are doing, especially hydration. Looking at urea, electrolytes, sodium. Okay, home parenteral nutrition. Let's look at indications, recommendations, the plans for discharging that patient, the teaching and monitoring. So indications and recommendations, and those comes from the ESPN guideline on home parenteral nutrition. ESPN says that those patients that are unable to meet their nutritional requirements via the oral or enteral route that okay, I'll start again. Indications and recommendations for home parenteral nutrition from ESPN guidelines. And what ESPN says is those patients that cannot meet their nutritional requirements through the oral route or enteral route needs parenteral nutrition support. So the kind of patients are those with transient reversible or the irreversible chronic intestinal failure. And we need to make sure that the patient environment, that the home is able to accommodate that way of feeding the patients safely. For cancer patients as well, those with chronic intestinal failure that have life expectancy above 3 months, where obviously the gut is not working, then parenteral nutrition is recommended. So plans for home parenteral nutrition, communicating with the patient, training, communicating with physicians in the community. There has to be a home parenteral nutrition hospital team or one in the community. So it needs to be carefully monitored. There needs to be a nutrition nurse involved. Suppliers, contact with the suppliers, nutrition goals. communication with other healthcare professionals as well, discharge plans, results of biochemistry. Biochemistry in the home parenteral nutrition, in parenteral nutrition, needs to be very tightly controlled. Any modifications to the parenteral nutrition regimen needs to be communicated with supplier, physician, and patients. And obviously for patients going on holiday, for the patient traveling, then they do need support with that. So prescription, the patient will need up to 3 days or so, or maybe even longer, depending on their tolerance. But we started parenteral nutrition with a continuous infusion over 24 hours. And gradually, and this can take up to 3 days, you reduce down to the cyclic mode of feeding, to feed over 12 hours. We need to consider glucose, and we know that 2 grams of glucose per kilo is the minimum that we need. Glucose is the fuel for the brain, but then that's maximal level as well, and we shouldn't go about 7 grams per kilo. You know, the high glycemic levels is associated with hyperglycemia, the fatty liver, you know, the foie gras syndrome that we talked in Europe, and respiratory problems. At the end of the cyclic PN administration as well, you might consider reducing that infusion rate, you know, halving that infusion rate for the last hour or half hour to prevent rebound hypoglycemia. Patient teaching, a lot of teaching here, managing the pump infusion. They obviously do need a pump here. Is spiking that home parenteral nutrition infusion, connecting, disconnecting the tube, communication links with the supplier. The patient needs to check that the PN that he's getting is really what has been prescribed for him, and how he's going to add multivitamins and medication if necessary. And then monitoring, extremely important. Nutritional efficacy, tolerance of PN. We need to check body weight, similar to enteral nutrition, hydration status, energy, fluid balance, biochemistry, really important. Nutrition is going through the vein. Biochemistry needs to be checked regularly. Full blood counts, albumin, looking at signs for infection, C-reactive protein, electrolytes, hydration status, kidney function, liver function, and glucose. And obviously look for signs and symptoms for vitamin deficiency or toxicity, and for trace metals as well. Okay, so something that I really want to talk about is about the impact of this emerging virtual care era that came to us, you know, following COVID. So this is data from UK, and in 2019 in the UK, 80% of consultations of medical interventions, they were face to face. With COVID, you know, 2020, with social distancing, then those consultations face to face came down to 7%. It has improved now. Obviously, we were coming out of COVID, but it's still around 50%. So what healthcare professional learned is to appreciate the benefits of virtual support, of virtual caring, and virtual monitoring for the patient. It's a solution for staff shortage. It's better for our patient. They don't have to travel long distance, and you know, care can be achieved at long distance. Okay, so one way that we can get the patient to work with us, the patient doing their own assessment and communication to the clinician virtually, is use the SARC-F questionnaire. That's the questionnaire that is predictive of sarcopenia. It's a questionnaire looking at strength, assistance with walking, rising from a chair, climbing stairs, and falls. Each parameter, as you can see, the score is from 0 to 2, and you've got 5 checkpoints. So a SARC score that is equal 4 or above is predictive of sarcopenia. And I quite like this cartoon mode. This is something that the patient can access virtually, or you can give the patient, and he can monitor how he's doing with the nutrition support, how he's progressing. Okay, so let's look at muscle mass strength and assessment. And a really good way to monitor patients, especially muscle strength in the lower limbs, is the gait speed 4 meter walk. This is really simple. What you need is the patient to walk 4 meters and measure the time. How long does it take for the patient to walk 4 meters? We should be able, healthy adults should be able to walk 1 meter within 1 second, or even less, really, 0.8 seconds to the meter. If our patient is taking over 5 seconds, this is predictive of disability, falls, mortality, and long-term care. Another very simple method that a patient can do at home and was developed by Tanaka et al., this Japanese group, is the finger ring calf circumference test. And we know that calf circumference has a very good correlation with muscle mass in the calf measured by CT scan. So you just form a ring with your finger and you measure the calf circumference, as you can see the patient doing. If it's bigger, then that is fine. If your ring just fits, progressing, it's okay, but it's not so great. Ideally, you want bigger. And if it's smaller, if your calf circumference is smaller, then it's a high risk of sarcopenia. So let's look at functional parameters now, looking at strength. And those muscle strength, they are correlated with muscle mass. A simple test is the get up and go test. Essentially, you want the patient to get up from the chair, walk 3 meters, turn back, and sit down again. And that exercise he should do within 20 seconds. More than that, again, demonstrate low muscle mass, low muscle strength. Then there is the widely used sit to stand test. Simple, you want the patient to sit down, hands crossed across the chest, and he's gonna get up and sit down. And he's gonna do that 5 times. So 5 repetitions of sitting and standing. And the patient should be able to do that within 15 seconds. Healthy adults should be able to do within 10 seconds. If the patient is taking longer than that, then it demonstrates low muscle mass, then low muscle strength. And then the hand grip dynamometer, a very inexpensive tool that a patient can buy and have those at home, and they can just see the strength of their hand grip. Less than 27 kilos for men is the cutoff point, and less than 16 kilos for women, a cutoff point, and is associated with low muscle mass. So in summary, my key message is that the right patient needs the right nutrition intervention. Delivery mode needs to consider the patient lifestyle, needs to consider the patient home environment. Teaching the patient caregivers is paramount for success, and appropriate monitoring will tell us if that nutrition support is effective, will support positive outcomes. Thank you so much. Those are my contact details. I'm on Twitter. That's my email. I will be answering your questions later on. Thank you.
Thank you, Dr. de Brito-Esserst, for your wonderful presentation, and welcome everyone to another Q&A session with some questions flying in from around the globe to discuss about. And I would like to start right away with the first question. With regards to digital monitoring and home parental nutrition, in the past, the patients were visited at home as often as they needed by the nurses. And nowadays, we see a different trend. It goes towards less visits, reducing the contacts, maybe also because of Corona, and using digital tools to monitor the patient. How far is this actually developed in the UK, and how does the future look like, according to your opinion?
It's still very much in its infancy. With the COVID, the pandemic, the whole virtual monitoring of the patient started, and you saw in my slide that in the UK, the face-to-face reduced considerably. So we've initiated that virtual monitoring using functional parameters, the sit to stand test, get up and go test, gait speed, but it's very much in its infancy. It's still being developed. It's not for every single patient, as you can appreciate. So no, it's very much in the beginning. Don't think we are far ahead, because we are not.
But I think the trend is going towards this direction. And as you say, it really depends on which patient you're probably seeing. Yes, absolutely. Well, traditionally, the focus for home parental nutrition therapy is normally on the short bowels patients. And is there a development in the UK to use this therapy also for the cancer patients?
Yes, but for the cancer patient, obviously, this is very much a discussion with the family, a discussion with the patient, looking at the quality, patient's quality of life, obviously, and looking at prognostic of the patient. So yes, I'm the head of therapy and rehabilitation for the Marsden Hospital, and we do use frequently for our cancer patients, especially when the intestinal failure is reversible and it's for a short period of time.
Okay, but you're also saying that it really depends on the patient case as well. You have to decide.
Very much so.
Case by case. All right. Well, and then there's another question. It's with regards to, patient empowerment. So are patient involvement and patient empowerment upcoming topics nowadays? And do you feel that patients question details of the therapies they're receiving? For instance, I prefer having my back during the nighttime. So can they actually interfere in the treatment?
I'm sorry, can you repeat the question again?
Yeah, sure. No problem, dear. It's about patient empowerment and patient involvement is this an upcoming topic? So for instance, do the patients actually question parts of their therapy? For instance, they're asking, can I have my back during the nighttime and things like that? So do they actually interact with the treatment they're receiving?
Yes, absolutely. It's something that it should be done with the patient. I said that the right treatment for the right patient, and that's really important when you're deciding the time to feed the patient. If it's going to be at night, it's going to be during the day. It depends very much on the patient lifestyle, on the patient preference. If you have somebody that is very active, then they might just want to be fed during the day. It's very much something that needs to be discussed with the patient. And patient empowerment, it's extremely important. I don't think we do enough of that. I think we still have our medical system is still a very patronizing one, that we want to do everything for the patient. But we need the patient to work with ourselves. We need to give some of that responsibility to the patient, and deciding the best time to feed, obviously within the clinical limits, is something that must be decided with the patient and with patient participation.
Okay, so you're also actually encouraging patients to get involved yourself. Does this also have to do with age, or the younger generation, or, changing generations over time? Because, the people today in their 60s, they're not the generations we know from 20, 30 years ago. And they're also using digital tools. They get their information, etc. So do you think also this is also influencing this development?
Yes, at the Marsden, we did with the pandemic, we changed our system, and we started introducing far more virtual monitoring of our nutrition services. And we did focus groups with patients, and was surprising a lot of older patients that, they thought computer wasn't for them, but actually quite enjoyed and thought that this is really cool. And they found out that it's not that difficult. So it was really surprising that a lot of our consultations now are virtual consultations. Even some speech and language therapy doing assessment to see if they can start eating now safely or not. They do that virtually, getting the patient to have a bit of water, observing if there is coughing, having a patient to have a bit of yogurt and checking if there is coughing or no. So a lot can be done virtually, and I wouldn't be put off by patients' age.
All right, good. Thank you so much for this wonderful insight. As I see, time's up already. So thank you so much for being with us in the Q&A session, Dr. Ioni, and for sharing your expertise with us in the presentation. I think we also have some time to take a refresher break. So I see you in about 10 minutes at 1:40 CET time. See you back then. Thanks.
Thank you.
Hello, everyone. It is 1:40 CT, the perfect time to move on with our blog on home artificial nutrition. After having heard a 21st century perspective on home parental nutrition, I would now like to open up the curtains for Dr. Katja Lanza and his speech on the importance of vitamin K on long-term home parental nutrition patients. Dr. Katja Lanza, please go ahead.
Good morning, and thanks a lot for the opportunity to participate in NutriTalks 2022. The topic of my presentation will be the role of vitamin K in long-term parenteral nutrition. And when you talk about long-term PN, immediately you think of chronic intestinal failure, which is a long-lasting reduction in gut function below the minimum necessary for the absorption of macronutrients and water electrolytes, such that intravenous supplementation of calories and all nutrients or simply fluids is required. So the two criteria to define chronic intestinal failure are the reduction of the absorption of macro and micronutrients and water, and the need of parenteral nutrition. The main cause of chronic intestinal failure is short bowel syndrome, which is caused by a surgical reduction of the intestine. And this is secondary to different diseases or clinical conditions like a bowel infarction, necrotizing enterocolitis, actinic enteritis, cancer, Crohn's disease, trauma volvulus. And certainly the classification of this syndrome is characterized by the anatomic intestinal peroresection residues. So we have patients with ileojejunal anastomosis, jejunocolic anastomosis or jejunostomy, and the less the intestine, the residual intestine, the worse is the prognosis, and more complicated is the management of this disease. And the main support, the main therapy for this condition is parenteral nutrition support, which can last for many, many years, and it can be also necessary for the whole life of these patients. And there are specific guidelines for the management of HPN, home parenteral nutrition. So we have established the range for calories, amino acids and lipids, and the fluids. But certainly the requirements of fluids depends on the single patient's characteristics, fluid losses, fluid intake, and compliance to therapy. So the management of parenteral nutrition in this situation should be personalized. And in particular, it should comprise all the nutrients, in particular, essential nutrients. And going to essential nutrients, so we now talk about briefly about vitamins. Vitamins are organic molecules or set of molecules. They are fundamental for proper functioning of metabolism. They are required in small quantities. They are essential because they cannot be synthesized at all, or they are synthesized not in sufficient quantities by the body. So they must be provided. And first vitamin requirements were summarized in PN, were summarized by the Food and Drug Administration in 2000 and were reviewed 3 years after. And in these guidelines, the vitamin K is recommended as other vitamins. Now there are the new European Society for Parenteral Nutrition guidelines, which are published online. They have been published, and they update the requirements for macronutrients and vitamins. there are other setting of patients which require vitamin supplementation during artificial nutrition in general. So going back to ESPEN guidelines for intensive care, it's stated that all PN prescription should include daily dose of multivitamins and trace elements. And also in the surgical settings, it's fundamental to provide patients with a full range of vitamins and trace elements. This is true for after surgery and for all patients. So it's very important. And wisely, the Food and Drug Administration underline that we should not wait for the development on clinical setting on vitamin deficiency before initiating vitamin supplementation. So we have to anticipate the need and we have to prevent deficiency. This is very true. And it is also stated by the new guidelines, which have been published recently, and they will be soon out and will be available for everybody. And we underline recommendation 1. I think it's the most relevant recommendation. So, which states that adequate amounts of essential trace elements and vitamins shall be supplied to all patients receiving medical nutrition from the beginning of the period of nutritional support. And if you notice, the grade of recommendation is A and the consensus in 100%. And among these vitamins, certainly vitamin K has a very important role. And we will go through that in a few minutes. The English colleagues of the BaPen 3 years ago notices very wisely, very interesting, that a patient with short bowel syndrome undergoing PN, HPN, were at risk of deficiency. They had a case of a patient with hemorrhage, with severe hemorrhage, and they noticed that there was a severe vitamin K deficiency. So they reviewed all the patients, they are more than 120 patients, and they noticed that the prothrombin time was longer than normal, and especially there was an association between this problem and the deficiency of vitamin K, which was provided by PN solutions. And so they stressed and they reinforced the concept that vitamin K is fundamental in patients treated with parenteral nutrition. Vitamin K function is fundamental for coagulation. It functions in the post-translational modification of at least 20 proteins via gamma carboxylation of specific glutamate residues to produce gamma carboxy glutamate residues. And gamma carboxylation is essential for activation of coagulation factor 2, 7, 9, and 10, but also for proteins C, S, and Z, which have anticoagulatory effects. So it's fundamental for the balance of coagulation. Interestingly, vitamin K dependent proteins have limited numbers of glutamic acid residues capable of carboxylation per molecule. So this is the reason why if you provide patients with high doses of vitamin K, you don't cause thrombosis or you don't enhance blood coagulation because of the saturation of the residues for vitamin K, and this is a very advantageous. There is not just coagulation, we also have a very important role for bone matrix mineralization of vitamin K. It works with the gamma carboxylation of osteocalcine, which is a peptide which regulates bone mineralization together in synergy with the vitamin D3. So we have a synergic role of vitamin K and vitamin D. And as for coagulation, the functionality depends on carboxylation status of this protein or osteocalcine. And so this balance must be maintained if we want to avoid and to prevent the risk of bone fractures of our patients. So according to what we are saying, main problems, the main signs of the vitamin K deficiencies are hemorrhage. So it's the predominant clinical sign. And if it's very serious, it can lead to fatal anemia. And we can detect it simply by blood test. But the important thing is to prevent it, because once it started, it's very problematic to manage and to treat. Of course, we can treat with high doses of vitamin K, but it can be very dangerous for our patients. And also bone fragility and enhanced vascular calcification must be prevented with adequate and timely provision of vitamin K. So which are the patients who are more at risk of deficiency? As we were saying, certainly they are patients undergoing artificial nutrition and specifically parenteral nutrition, and especially patients treated with antibiotics or anticoagulants. Antibiotics affect the intestinal flora, and so they can reduce impact the endogenous production of vitamin K. And certainly the traditional anticoagulants are agonists with vitamin K. So at the liver level, they block the enzymes which act together with vitamin K to activate coagulation factors. So vitamin K is fundamental for the stability, for the balance of the coagulation process. Luckily, new anticoagulants are not vitamin K dependent, so these problems can be reduced or prevented as well. What should we do with parenteral nutrition? Certainly, there are lipids which contain a certain amount of vitamin K, but if we go to see the amount, so we realize that it's less than 1/5 or 1/5 of the recommended daily dosage of vitamin K, which is 150 micrograms. So it can't be sufficient with the simply with the 3 chamber bags. We are not giving patients the correct amount of vitamin K. And we also have to take into consideration that there is a high variability among patients, which depends on medications, on clinical conditions, and on oral intake of nutrients. So we really have to take this into consideration. Nowadays, there is a new product which is available in the European market, which we are testing and we are using in our hospital with success, with no problems. And so now we are satisfied because we are aware that we are providing the right amount of this important vitamin to our patients. And this product now reflects 100% of what was recommended. It has been recommended by the new ESPEN guidelines regarding the provision of vitamins and micronutrients in artificial nutrition. So to summarize my talk, I would say that not just patients with intestinal failure, but also all patients receiving parenteral nutrition in important amounts for satisfying their nutritional needs are at risk of vitamin K deficiency if they are not appropriately and timely provided with vitamin K administration. There are vitamins in the market, and now there are these new products which respect the ESPEN, ASPEN, and FDA recommendation. So we have to remember to add vitamin preparations to parenteral nutrition, and among the vitamins, we have to be aware of the importance of the correct and appropriate dosage for adults of vitamin K, which is 150 micrograms. I thank you for your attention, and I will be certainly available for Q&A during the event.
Thank you for this great presentation, Dr. Katja Lanza, and showing us the importance of vitamin K in long-term home parental nutrition patients. And thank you for joining this Q&A session now, even from the train you're on, visiting probably or doing important business trips. So I'm glad you're joining us from there. I hope you can hear as well. And I'm happy to tell you that we have received a couple of questions regarding your topic, which I would like to discuss with you. So if you're ready, we can just start right away with question number 1.
Yes, I'm ready. Thank you.
Great.
I hope we can go on with connection. Okay.
Sure, we will see. That's the, the thrill of having a live session. Well, the first question is, does excessive vitamin K intake result in overcoagulation, and does it increase thrombosis risk?
Yes, the answer is simple, because it's no, because we have proteins linked to vitamin K activity are saturated. They have limited domains which can be binded by vitamin K. So the excess in an increased risk of thrombosis and an increased. So it's safe, and because the excess does not act with the proteins, because it doesn't bind to the active sites.
All right. That's a clear statement. Thank you so much for that. And you have mentioned it very briefly, but could you explain again a bit about the vitamin K content, which is available in the lipid emulsions? Does this supply the recommended 150 micrograms per day?
No, average is 1/5 to 1/7 of the recommended dosage. So it's not enough at all. It's 20 to 30 micrograms, while the recommended intake is 150 micrograms. So it's not enough at all. So we have to supplement it.
All right. And is the constant level of vitamin K important? And if yes, why is that like that?
Sorry?
Is the constant level of vitamin K important? And if yes, why is it important? Did you read the question? I guess we have a frozen picture. Ladies and gentlemen, just stay tuned with us.
So there are some mild problems with the question. Can you repeat the last question, please?
Yes, of course. No problem. It's absolutely no problem. The constant level of vitamin K, is that important? And if it is important, why is it important?
It's important for coagulation balance and also for the bone health. So we have to be able to maintain constant levels and to supplement patients with the correct amounts of vitamin K, especially if patients are fed exclusively by parenteral nutrition, because they don't have any other source.
Okay. All right, so we need it, and we need to supplement if it's not sufficient. Okay, we got that.
We need every day, we need the right amount of vitamin K.
All right, good to know. How much vitamins would you recommend for home parental nutrition? For instance, daily, 3 times per week, or what do you recommend?
Well, if patients are fed exclusively by parenteral nutrition, is supplemented on a daily basis. If parenteral supplemental will be balanced, but we have to check blood tests. We have to dose vitamin K, and then you can adjust the doses according to the blood test results. But the more is the dependence on parenteral nutrition, the more is the need for daily supplementation of vitamin K.
All right. So I got that right. You recommend a daily supplementation, respectively, really to check on the patient according to blood tests if there are deficiencies, etc. Did I get that right?
Yeah.
Okay. Good. Thank you so much. And what is your personal experience? Do patients on home parental nutrition actually receive enough vitamin K? So from your own practice.
From my own practice, things could be improved because we have limited access to complete vitamin supplementation. It depends on the tenders and on the different choices. Now that there are new products, I think things should be taken into consideration better, much better, because many times the problem is that the vitamin K insufficiency is subtle, which sometimes cannot be clinically so clear. But we have to force us to think in perspectives and to prevent problems rather than to treat them when they occur. So, in my view, we should take much more into consideration possible deficiencies, and vitamin K is a very important.
Yes, well said, Dr. Kaczylec. I hope you're not losing the connection. Let me just quickly check once again. Can you still hear us? Seems like that we lost the connection. That's why I think we have a couple of questions left, but I think we will leave that to the expert session later on. Can you still hear us, Dr. Kaczylec?
No, I now I can. I can hear you now.
Okay, and we can hear you. Then I'd like to take the chance to place a final question to you. So thank you for the statements before. And this is with regards to IV lipid emulsions. They mainly contain considerable amounts of fat-soluble vitamins, for instance, 200 milligram per liter emulsion. And therefore, is it safe to add additional fat-soluble vitamins to nutritional regimens, like especially in the pediatric patients?
No, sorry, can you repeat it? I have some problems.
Yes, of course.
There are classical lipid emulsions?
Exactly. It's about the lipid emulsions, which already contain a lot of fat-soluble vitamins. And so the question is towards especially pediatric patients, if it's safe to supplement if there's already a considerable amount of fat-soluble vitamins in the lipid emulsions, especially in pediatrics.
You should also think of dosing. Pediatric patients are more particularly fragile, and metabolism is particularly intriguing, and I think you should really plan to dose vitamins at least in the early phases of parenteral nutrition, and then to reassess blood tests. In general, these lipid emulsions, as we were saying, are not rich in vitamin K. So it's important that you take blood tests and you assess the adequacy of supplementation, also particularly in pediatric population.
All right. So you're saying it is safe unless or at least if monitoring is done of these patients, then it's also safe.
Definitely.
For sure. Well, then having a look at the time, thank you, Dr. Kaczylec, for your time, even on the train, and your contribution, your presentation.
I'm sorry for the inconvenience, and thank you very much.
We have to thank you. And we are again proceeding into a short coffee break now, 10 minutes prior to our next block, the fourth one of today, nutritional support in specific clinical conditions. And I'll see you in a few minutes. Thank you. Hello, welcome back. It's 2:30 p.m. CT, and I hope everyone has had the chance to shortly re-energize for our fourth round of today's NutriTalk event, now dealing with the topic of nutritional support in specific clinical conditions. And within this block, it's my pleasure to introduce you our three next speakers, Dr. Philippe Fouquet, Dr. Danielle Fumelli, and Dr. Ben Weise. Dr. Weise is a medical doctor specialist in internal medicine, nephrology, and nutrition care. And having more than 20 years of clinic experience in hospital and outpatient care, he joined b.braun as a medical manager in nephrology in the MSA Affairs Department. Today, Dr. Weise will share with us intradialytic nutritional strategies with b.braun product in kidney disease patients. Dr. Danielle Fumelli is a doctor of clinical nutrition and dietetics at the Hacienda Ospedaliero Universitaria in Ancona, Italy, with a broad expertise on the field of nutrition and oncology, metabolism, and artificial nutrition, as well as research and education programs. Hence, the topic he's going to present to us today is focused on nutrition in oncology and target medical nutrition experience, the use of omega-3. Dr. Philippe Fouquet is a nutritionist doctor who had led the largest French clinical nutrition unit in Cannes, welcoming patients with severe digestive pathologies, chronic wounds, or severe obesity. And today, he is a consultant and develops nutritional trainings dedicated to home care, teaches students in dietetic schools, and trains dietitians. We will learn from Dr. Fouquet how malnutrition in patients with ostomy can be prevented, and secondly, how healing in patients with chronic wounds can be improved. Again, I would like to motivate you, our audience, to post your questions, your comments and remarks throughout the session, and we will be happy to come back to them in our question and answer session later on. But now, Dr. Fouquet, the stage is yours.
Okay, thank you for attending this presentation. Today's presentation is about the importance of nutrition for patients with ostomies and chronic wounds. After a brief introduction, we will focus on the metabolism and more specifically about the process of energy production. Then we will see two key points to optimize patients' diets. And then we will finish by the specific diets for patients with ostomies and chronic wounds. Let's start with the digestive tract. It's the organ of food digestion, meaning it transforms solid food into nutrients, and those nutrients, of course, will be absorbed through the intestine wall. But it's also the organ of liquid absorption. It absorbs all the liquid we drink and also all the liquid produced by the digestive system. For example, we produce every day about 2 liters of saliva. The stomach produces 2 liters of hydrochloric acid, pancreas 1.5 liters, bile 0.5 liter, and even the small intestine produces 2 to 4 liters a day and secretes also plenty of salt. So ostomies will decrease absorption by reducing the absorption area because the length of the intestine is shortened by the surgeon, by increasing the progression of the nutrients in the small intestine because there is no longer, I would say, the feedback for the control of the velocity of the nutrients. And some enzymes are damaged, for example, lactase. Lactase is a very fragile enzyme, so patients with ostomy won't be able to digest milk anymore. So ostomy is increased risk of undernutrition because less nutrients are absorbed. The healing process is delayed because of the lack of nutrients. Same thing for infections. Less liquid absorbed, so high risk of dehydration that can lead to renal failure. And less salt absorbed that can lead to hyponatremia and coma. The list could be longer, and we have also risk of anemia with the lack of absorption of vitamin B9 and B12. So I could say that the first medication for patients with ostomy should be food. Now let's see the problems with chronic wounds. Let's take an example of a woman, 65 years old, 60 kilos, and 1.65 meters high. Her basal metabolic rate, it means her energy needs for one day is more or less 1,250 kilocalories. I did use the Harris and Benedict formula to calculate this. If this woman does some physiotherapy, you need to add 10% to these energy needs. But if she has a chronic wound, you will need 30% more. And if she gets a systemic infection, maybe due to an infected chronic wound, you will need to add 60% more. So, food must bring all the required energy for the healing process. That's what we are going to see in this chapter. How can we produce energy? Just let me remind you all the nutrients. We have the macronutrients, which are the carbohydrates with complex carbs and simple carbs. Proteins, it's a combination of amino acids, and lipids, it's mainly fatty acids. On the other hand, we have all the micronutrients, which are vitamins and minerals. So those nutrients are used mainly as body building block for cell renewal, for example, for the healing process, substance production, for example, insulin, hormones, enzymes, and I would say for all the organ functions. Only 3 of the nutrients, the macronutrients, are also used as fuel to produce energy. And we are going to see what are the 2 processes for energy production. By the way, I would like to point that vitamins and minerals cannot give energy because they cannot be used as fuel. It means if in the morning you feel tired and you eat some vitamins and mineral pills, and after that you feel better, it's only the placebo effects. So let's see the main process of energy production. We have the 3 macronutrients, proteins, amino acids in the muscles, and fatty acids in the adipose tissue. Glucose is widely used in some glycolysis reaction to produce energy. Then we use also fatty acids that must be combined to glucose to enter in the Krebs cycle. The Krebs cycle is the most efficient process to produce energy. It means with very few quantities of fatty acids, we can produce lots of energy. And in this process, we don't use amino acids. The other process, the accessory process, we have our 3 nutrients. In this process, we use only very few quantities of glucose, and we will see why just after. It's only for specific cells as brain cells, heart cells, or renal cells, for example. In this case, fatty acids cannot be combined with glucose, so they will be transformed into ketonic acids, and this ketonic acid will provide energy. But the problem is the human body doesn't like to have a high level of ketonic acid, so we can only transform small quantity of fatty acids. So amino acids must provide the energy required in some catabolic reaction. The key point of the energy process production is how can we activate the main process instead of the accessory process. In the main process, you can easily see that patients are not losing weight because they are not losing muscle. And in the accessory process, they lose weight by losing muscles. The answer is we have very few stocks of glucose. It's almost 300 grams for an adult. So food must bring everyday glucose into our body. Well, and besides, as food in the accessory process doesn't bring glucose, you can see that we need to produce glucose from amino acids. So in the accessory process, patients are losing lots of weight. What are the source of glucose brought by the food? Sugar. The problem is sugar comes quickly and goes out quickly too, 30 minutes, more or less. But starches will be able to bring glucose for 5 to 6 hours into the bloodstream, and bread, it will be during 2 or 3 hours. You could say, okay, I'll prefer sugar, so I will eat ice cream, for example, every 30 minutes. But the problem is a spike of glucose into the bloodstream will be transformed into fat, and so it won't be able to produce energy. So, to activate the main process, the answer is simple. Eat complex carbs, eat starches. Now let's see 2 key points for an optimal diet. The first key point is irritating foods. Some ingredients irritate the gut mucosa. The first one is a tea and herbal tea. It reduces the tonicity of the lower esophageal sphincter. It also increases frequency of crisis for patients with Crohn's disease. Second type of irritating foods, it's raw vegetable and fruits and whole starches because they contain non-hydrolyzed fibers, so it produces lots of fermentation. Third one, milk and yogurt, as I said previously. The lactase is damaged after an ostomy, so patient won't be able to digest milk and yogurt. So into the small intestine, you will find lots of lactose, and bacteria love lactose, and they will produce lots of gas and even diarrhea. Last one, sparkling beverages, because bubbles, the CO2, reacts with the hydrochloric acid to give carbonic acid, which is very, very irritating. What are the consequences of irritating food? It increases the outputs because of the inflammation of the gut mucosa. It produces lots of bloating and pain because of the gas production and more peristalsis movements. And it provides undernutrition because the chyme, which is the mixed nutrient into the small intestine, moves faster because of the diarrhea, for example, so less nutrients are absorbed. Let's see the volume of the output with eating irritants. And without eating irritant for jejunostomy, we can almost divide by 50%. The same thing for ileostomy. Second key point, how many times should a patient eat starches? The answer is 3 times a day. For example, in the morning, bread will bring glucose during 2 to 3 hours. And for lunch and dinner, rice, noodles, potatoes will bring glucose more or less for 5 hours. So starches on the plate for breakfast, I would recommend 75% of the plate with starches. For lunch and dinner, 50%. And in the afternoon, if the patient feels hungry, give him 75% of starches in a small snack, but don't give him a fruit or a yogurt, of course. In some countries, people prefer to eat a larger breakfast, so in this case, 50% of starches is enough. And for lunch, I prefer to have a snack, but this snack must have 75% of starches. So now let's see the specific diet for patients with ostomy and chronic wounds. For a patient with chronic wounds, key point, energy. So they must eat starches 3 to 4 times a day. Then you can add some bodybuilding blocks as proteins, or if the patient doesn't eat enough, you will give him some food supplements with proteins. And you can add fruit or vegetable. They contain lots of vitamins and minerals, or pills of vitamins and minerals, if they don't like to eat vegetables and fruits. Patients with jejunostomy or proximal ileostomy. Non-whole starches 3 to 4 times a day for energy. Of course, no irritating food because they have already high outputs. Add proteins. Vegetables are not necessary because they have no more colon. Mainly, they will need parenteral nutrition and hydration and parenteral vitamins and minerals because they cannot absorb enough nutrients. And they will require a monthly shot of vitamin B12 because vitamin B12 is absorbed only at the end of the ileum. For distal ileostomy, guess what? Starches 3 to 4 times a day for energy. No irritating food because of the high outputs or risk of high outputs, add proteins and cooked vegetables. If the patient is losing weight, you can add food supplement or enteral nutrition, or if he doesn't support enteral nutrition, you will set a parenteral nutrition. And of course, vitamin and minerals supplementation, and to see if he needs vitamin B12, just do a blood analysis to check the blood level of vitamin B12. For colostomy, starches again, 3 times a day at least. More or less irritating food, depending on the consistency of the stools. Add protein and vegetables, food supplement or enteral nutrition if he's losing weight, and vitamin and mineral supplementation, of course. For urostomy, starches, now you know we need starches 3 to 4 times a day. No excess of irritating food because it may cause lots of bloating and it will press all the urinary system. Proteins and vegetables, food supplement or enteral nutrition if they lose weight, and vitamins and mineral supplements. 3 weeks after an ostomy surgery, first step, please don't give them tea, only coffee. And if they don't like coffee, it's going to be lemon water or water with syrup. Second step, you can add some fruit puree, but no dairies, no milk. Third step, you can give them blended balanced diets. It's going to be mainly potatoes puree. And of course, no irritants and no vegetables. And the next step, you can add vegetables if possible, for example, for distal ileostomy or colostomy. To conclude, the irritating food, you must adapt to the type of ostomy and to the volume of the outputs. Starches at least 3 times a day to produce energy. Nutritional support with food supplements or enteral or parenteral nutrition. Parenteral nutrition will be needed for jejunostomy or proximal ileostomy. Vitamin and mineral supplementation, and pay attention to vitamin B12. And of course, hydration for ostomies with high outputs. I'll thank you for your attention, and I will be ready to answer all the questions.
Thank you for these great insights, Dr. Fouquet. Well, I've seen a lot of questions coming in during your presentation, and so I would like to directly pass the first one to you to start our question and answer session. Would you recommend a nutritional screening and, if needed, an assessment for all patients with ostomy and or chronic wounds in the hospital? And if yes, which tools would you recommend?
Well, I would say it's not necessary to do nutritional screening as the diet will be modified after the surgery or after having the chronic wounds. So I would say we can ask the patient what is used to eat to just fit with the diet we will recommend to him. So not a real screening to do.
Okay, so this is the difference actually in compared to hospitals, that you would not recommend this on a regular basis. So it really depends on patient as well.
Yeah.
Okay, but to be honest with you, when you say you would ask the patient what has he need, it's like a screening you do, right?
Yeah. A small one.
It's a small screening. All right, good. Thank you so much. And we have another question. Do you think that nutrition can be used as a standalone therapy, or can it change in the lifestyle, support the wound healing process? You know, physical activities, maybe stopping to smoke or prevention of diabetes, things like that.
With nutrition?
Yes. So is it a standalone, or can you actually have other factors that the patient can actually promote in order to promote wound healing, respectively prevent the development of these kind of wounds?
Of course, if the patient is able to walk a little bit, do some physiotherapy, it will be better for the healing process compared to staying in bed all the time.
Okay. So you would not say that nutrition is a standalone therapy. So it's like also other effects. It's one of the major factors that should be looked at, but also the others. Okay. And what about other risk factors? Smoking, diabetes, all that?
For the healing process, my experience showed that the most problematic thing is alcohol, more even than smoking. But smoking is bad, but alcohol is worst.
Interesting. Oh, I didn't know that either. So, all right. Okay. Thank you for that insight. And there's the last question I have here. Would you also recommend the supplementation of single macro, respectively micronutrients as well, instead of an individualized dietary plan? For instance, the supplementation of zinc, selenium, proteins alone.
I would recommend at least to give a micronutrient supplementation, because we don't know the quantity of micronutrients in the food we are eating, or even in the parenteral bags with vitamin supplementation. So I think just to be secure, it's better for patients to have some micronutrient supplementation, but no need for proteins or fat or starches.
Okay, all right. Well, then let me have a look at the chat once again if we have any further question. Doesn't seem to be the case. And with that, I would like to thank you once more for joining us today. And we will go into another short break, allowing our audience to refill their drinks or to grab a snack. Open the window. Maybe we will do this as well here. Do some stretching, whatever you prefer. And we'll see each other at 3:10 p.m. CT.
Thank you.
It is 3:10 p.m. CT. I hope everyone is back on board for us to directly continue sharing expertise within the context of services along the patient pathway. It's my pleasure to welcome Dr. Ben Weise, who will share his insights with us on the topic intradialytic nutritional strategies in kidney disease with Evo Forte, Nutriflex, and NutriComp. Dr. Weise, please go ahead.
Dear ladies and gentlemen, I'm very grateful to have the opportunity to talk to you about nutrition in kidney disease. Most of you already had the chance to have a look at the b.braun nutrition portfolio, and most of you will have recognized that we have a very broad range of products dedicated to patients with impaired kidney function. But what is the background behind this mission to provide such a comprehensive b.braun nutrition portfolio for patients with kidney disease? If we go to the literature and we go back to the very beginning of treatment of kidney disease, you can find this publication by Pendar's, where he describes his clinical experience with 16 patients he has put on chronic hemodialysis. And what he describes in the early 60s is mainly a severe state of malnutrition in his patients. The patients suffer from deficiency of thiamine, pyridoxine, vitamin B12, hypercalcemia, rib and hip fractures, nausea, vomiting, and much more. If you then go to the more recent literature, you see that hardly anything has changed. Malnutrition is, according to this publication from 2020, has a global prevalence of 1/3 to 1/2 of all patients, and the patients face a very high risk of mortality. In the literature, you find many different terms describing the state of malnutrition, and it lasted up to 2008, when the first harmonized nomenclature and the first harmonized diagnostic criteria for malnutrition were evolved. And what is striking is that malnutrition is, in this expert panel consents, mainly described as the occurrence of loss or wasting of proteins and loss and wasting of energy. And the header of both is kidney disease wasting. And that is because this protein energy wasting can occur in acute kidney diseases, but also in chronic kidney diseases. And remarkably, nearly all stages of kidney disease are affected. You see here in this table from Amparo that CKD 3-stage patients with moderately impaired kidney function, already more than 10% of these patients suffer from severe protein energy wasting. And with increasing severity of the kidney disease from stage 3 up to stage 4, the severity of the malnutrition state is also increasing. This is not only a localized problem in, let me say, less developed countries. No, It's a global challenge we face. And this, we do not have data from all countries, but those countries who provide data show that to a varying degree, but all in all, almost half of all kidney patients have a severe state of malnutrition. And going back to Germany, you can say that from all patients with chronic kidney disease, not yet requiring renal replacement therapy like dialysis, there are 800,000 patients in Germany requiring nutrition therapy, 800,000 patients who are malnourished. Additionally, you have 42,000 patients on hemodialysis, those patients with severely impaired kidney function, that need nutrition therapy. These are tremendous numbers. For sure, the question pops up, what is the background? What is the reason why so many of the kidney patients suffer from malnutrition? And not to go too much into detail, you can say that the kidney disease itself is the main trigger. If you have kidney disease, you accumulate uremic toxins in your blood. And these uremic toxins can cause chronic inflammation. And this chronic inflammation is a state where protein and energy is consumed. Moreover, uremic toxins cause a lot of metabolic and hormonal derangements that all contribute to malnutrition. Second, you have the treatment of the kidney disease. And who has already treated kidney patients knows they have an enormous pill burden. Some of the patients take up to 20 pills a day. And of course, this pharmaceutical therapy also contributes to the malnutrition state of these patients. Moreover, kidney patients have to stick to a very strict so-called kidney diet. And this kidney diet, of course, contributes to malnutrition. Most of those patients need to have their therapies in hospital, and the patients suffer from frequent hospitalizations, and all those hospitalizations contribute to malnutrition as well. If a patient is in the state of hemodialysis, of course, hemodialysis is beneficial in the sense that uremic toxins are removed from the blood of affected patients. But not only uremic toxins are removed, also protein, small molecular weight proteins, and amino acids are removed, and energy is removed, not only as amino acids, but also as glucose. So the dialysis therapy itself also contributes to malnutrition. This has, of course, impact on the outcome of these patients. And you see in this graph different stages of malnutrition for patients in the stage CKD 3 to 5 not requiring renal replacement therapy. And Aparo says half of all CKD patients with protein energy wasting have died within 42 months. That means 3.5 years. Patients with impaired kidney function not requiring dialysis, 50% of them have died within 3.5 years if they are malnourished. The situation for dialysis patients is even more severe. Half of all dialysis patients with protein energy wasting have died within 26 years. which is a little bit more than 2 years. It's not only a question of survival and mortality. It's also a question of morbidity. Of course, a patient with kidney disease and malnourishment has a higher disease burden than a well-nourished patient. And what you can say is they require more hospitalizations, and their hospital stays are longer. Also, again, contributing to more severe states of malnourishment. And last, not least, this has, of course, a dramatical impact on the quality of life of these patients. Malnourished patients have a much lower WHO score on quality of life than well-nourished patients. But there's hope. And I citate from Campbell. And Campbell says, providing individualized nutritional counseling improves many components of quality of life compared with standard nutrition care in the stage prior to dialysis treatment. Of course, this knowledge from Campbell and from all these studies found way into the guidelines. And I give you just a flavor of the guideline. I cannot show in this presentation the whole guideline or summarize the guidelines, but in crude, broad view, you can say that the clinical practice guidelines are structured in 5 steps. And the first of all is the step 1, the awareness creation. This awareness is an essential requirement not only for healthcare workers, but particularly for the patients. Patients with early, mild to moderate CKD need to know that they are threatened by malnutrition and by the malnutrition sequela. They need to know that their nutritional status needs regular assessment, even if they feel well nourished, just to make sure to intervene in the very earliest stages of malnutrition. What is indispensable, though it did not find way into the guidelines, is the evaluation. The evaluation after having started any nutritional therapy. Doesn't matter whether it was only counseling or whether it was a treatment with oral or intravenously supplements. The evaluation is so important because it shows to the patient that the therapist is questioning his own therapy. And because it explains to the patient, the therapy may work, but also may not work. And I will check whether it works. So and then one can switch and change the therapy approach and try to go another way. And this therefore, the evaluation is something that tightens the connection between the doctor, the physician, and the patients. And it contributes that nutrition therapy is always a joint approach of many stakeholders. So the evaluation plays a major role in practical medicine, though it is not recommended or, let me say, provided by the guidelines. But if you have now a look at the guidelines, you see that most guidelines have a low evidence level. You do not find evidence level 1A recommendation. No? And you see that even the authors of the guidelines are not very confident in the main recommendations that these recommendations work. And you have a second aspect. We learned already that malnutrition is a multifactorial process. And one factor may play a major role in one patient, but a minor role in the other patients. And other factors, they can affect or have an effect on each others. So what I want to say and explain is a standardized treatment approach is not possible because we have 2 weak guidelines that allow a standardized approach, and because malnutrition is so multifactorial. What we need is an individualized treatment approach, a treatment approach that is customized to the single patient. And I show you my proposal. And this proposal is very strict connected to the steps of the guidelines. So you start with the awareness. Awareness means to create awareness for legacy and policymakers, for clinicians, for patients, for all stakeholders. This is the step 1. Step 2 is then the assessment. And in this assessment, you assess the patient not only on demand, but on a regular basis, meaning each and every patient is assessed on a regular basis, no matter whether you think he has nutrition or not. And then you do the counseling. And the same refers to the counseling. Counseling must be provided to each and every patient, even those who are well nourished, because you want to protect them with your counseling from being malnourished. And then the 4th step is the treatment. And the treatment must be individualized and multimodal. It must be customized to the single patient. And the last step is then the evaluation. You question your therapy, you evaluate whether it works, and then you start the circle again. And this therapy approach is not only individualized approach, but a continuously running approach that is always running. Clear. This approach requires resources and it requires expertise. This approach requires treatments by professionals who have a high experience in this matter. And this approach requires a lot of resources. You have to go to each and every patient and counsel them and assess them and re-evaluate what it is. This is, in my eyes, only possible if it is outsourced from the standard kidney care. I think that the nutritional therapy of those patients must be outsourced. And possibly we find a service and we can develop a service called nutrition and kidney disease. And if this service is outsourced, we save the resources of the renal care centers because they do not have to spend effort, time, and money in the nutritional support of their patients because it is outsourced. So, all in all, I want to thank you and give you the following take-home message for you. Malnutrition in kidney disease is long known and detailed characterized. It is the source of tremendous suffering, morbidity, and mortality. B.Braun is committed to protect and improve the health of people all around the world. We offer a broad portfolio dedicated for this patient group, and we offer strong expertise to support you in fighting malnutrition. We are convinced that by sharing expertise between B.Braun, between B.Braun, policymakers, healthcare professionals, and patients, we can realize remarkable achievements. So let's go on and let's fight malnutrition. Thank you. Thank you for having the opportunity to talk to you.
There are sure questions, Dr. Weise. Thank you for your presentation, first of all. And therefore, let's directly jump into the first questions that have reached us. Knowing that renal disease patients are suffering from malnutrition at a very early stage of their disease, wouldn't it make sense to start nutritional counseling as well in the very early moment in their treatment pathways? What do you think?
Absolutely. I absolutely agree. That makes great sense. As I have shown you the data, the malnutrition starts in the very early stage and aggravates during the disease. It is very difficult to treat severe malnutrition. However, if you start the treatment early, I would recommend regular testing at least from stage CKD3 on. You can prevent the severe stages of malnutrition. So I agree, and yes, the answer is yes.
Okay, so early intervention as early as possible in order to prevent worsening of the situation. All right. Well, then there's another one. Can the electrolytes in the nutrition regimen harm the patient during intradialytical parenteral nutrition, or do we always need a special electrolyte-free compounded bag?
I spoke about individualization of nutrition therapy. So you cannot say that NSAID renal disease is always the same. Some patients have severe problems with their phosphate levels. Others have problems with their potassium levels. Others with their fluid intake. So I would say you have to assess the nutritional status of the patient you want to treat, and then the compound, the compound bag helps you to adapt the fluid you want to administer intravenously to the individual condition of your patient.
All right. So we can see that this very sensitive patient group here we are talking about, and that you really have to adjust as closely as possible with the patient conditions here. All right, got the message. And the final question we have here in the chat is, how do I connect the nutrition solution at the machine, and what accesses are needed? So should we use actually the arterial or the venous axis? Or is the separate access needed for nutrition, and how to connect all the dialysis machine? Maybe we just sort this question out. Let's start from the beginning. How do I connect the nutrition solution at the machine, and what accesses are needed?
The main advantage is that the patients are connected to the machine and that there's no need to do any extra needling to the patients. And you connect the bag on the arterial side, always on the arterial side of the system. And often there are questions Are there nutritional compounds that are lost by the filters? Yes, they are. However, as the blood is running continuously through the system, you always have the problem. And it is one of the main problems of dialysis without supplementation by intradialytic nutrition, that you lose energy and small molecular weight proteins during dialysis process.
So in other words, there's no need for an separate access for nutrition.
No,
so you're just the arterial side. And the last part of that question was how to connect everything to the dialysis machine.
You connect it to the arterial line of the dialysis machine.
And that's it.
There are connectors in our lines we use for our dialog machines, and you can use these connectors, and they go via Lure lock, and this is pretty easy and no problem at all.
All right, and I'm sure b.braun will always help out with their sales reps and representatives in the various countries to assist on that, of course, because we have the technicians there. Well, then I think we don't have any more questions here. Then thank you, Dr. Weise, for this great session, for your presentation and the question and answer session.
Yeah.
We are going to have another break now, and we'll meet you all at 4 o'clock, so 4 p.m. CT, back to the next session. Thank you.
Thank you for the invitation. Bye-bye.
It's 4 p.m. Central European Time, and I would like to welcome Dr. Fumelli, who will now share with us a presentation on nutrition in oncology and target medical nutrition experience, the use of omega-3. Dr. Fumelli, the stage is yours.
A warm greeting to all participants, and thanks especially to b.braun for their kind invitation. Many patients with oncological disease have a poor nutritional status, which has a negative impact on clinical outcomes. Malnutrition related to cancer mainly manifests with a serious reduction of the lean mass, which can occur in any phase from the curative to the palliative, and often coexist with obesity. We will discuss the opportunities related to the role of nutrition and in particular of omega-3 in the prevention of loss of lean mass and, if possible, in the reverse of the trend of the weight loss. Patients with oncological disease have a particular high risk of malnutrition, both to the mechanism of the disease and to the effect of their treatments. We know that the deaths from 10 to 20% of cancer patients can be due to malnutrition rather than to malignancy itself. According to some reviews, frequency and severity of weight loss vary according to the type of cancer. From 50 to 80% of patients with cancer of the upper gastrointestinal tract and 30% of those with extraintestinal cancer have weight loss at the diagnosis. In addition, we know that weight loss gets worse with the progress of the pathology. Premio, which means prevalence of malnutrition in oncology, is an Italian study that focused the nutritional status of almost 2,000 patients at the first oncological examination. This study has shown in a certain way that malnutrition, weight loss, loss of appetite, anorexia are common in patients since the first stages of the disease and already detectable at the first visit. 50% showed a variable degree of nutritional compromise. Of these, 40% were at risk of malnutrition and 10% were clearly malnourished. This study confirmed that the severity of malnutrition is related with the location and the stage of cancer. Furthermore, it was shown that in the first 6 months, 65% of patients lost weight in a significant way. The weight loss results in greater toxicity of the treatments, greater clinical complication, lower quality of life, and reduced survival. In this recent consensus, cancer cachexia is defined as a multifactorial syndrome characterized by a progressive loss of muscle mass, with or without loss of fat mass. That leads to a progressive functional loss up to a fatal compromise. Physiopathologically, is characterized by a negative balance of protein and energy, guided by a variable combination of reduced and altered food intake. In this recent article, the classification of neoplastic malnutrition is classified in a more complete way to have an optimal management of weight loss. Weight loss is driven by metabolic changes due to the cancer-associated pro-inflammation cytokines. The understanding of this mechanism certainly represents a challenge for the management of malnutrition. Once the patients with cachexia have been driven according to these criteria, and not only through the parameters of variation of BMI, it could be possible customize a multimodal therapeutical plan, including nutrition, pharmacological strategy, and physical exercise. This review shows the consequences of malnutrition in patients with cancer. Weight loss increases the toxicity induced by radiochemotherapy, worsens the sensitivity of tumor cells to anti-neoplastic treatment, weakens the defenses of the organism and increases the frequencies of hospitalization and the post-surgery complications with a lower quality of life that results in an increased mortality. In cancer, pro-inflammatory cytokines stimulate the degradation of proteins in the skeletal muscles, which results in a massive infusion of amino acids in the circulation to the liver. And where they are used to support gluconeogenesis and also the synthesis of protein in the acute phase. Also, the mass of the fat tissues is consumed due to the activation of lipases. The liver is used as a gluconeogenic substrate. The recycling of the lactate activates a cycle of Coris characterized by an high energy inefficiency. Alterations of the metabolic path lead to alteration of the metabolism and proteins and amino acids, with activation of apoptosis and reduction of muscle regeneration, while a decrease of EGF-1 suppresses the protein synthesis, also causing an increase in expression of genes related to mitochondrial disconnection and energy dispending. Third phenomenon is the browning of the white fat tissue in cachexia, favoring the production of heat and energy inefficiency. We consider cachexia as a multi-organ syndrome, in addition to the cachectic process. In fact, there are anomalies of cardiac function, alteration of the hepatic protein, and alteration of the central neurological system regulational functionality. Another very important mechanism that leads to malnutrition is dysgeusia, caused by drugs that have a neurotoxic effect and alteration due to the chronic inflammatory state. This study analyzes a sample of 1,199 cancer adult patients under chemotherapy recruited by 7 cancer centers. Descriptive analyses were conducted to estimate the prevalence of changes in diet, and data were used to examine the association between this alteration and health outcomes. About 40% of patients reported a reduced appetite since the beginning of treatment, and more than 60% of patients reported at least one chemiosensory alteration. The most common alteration described were the increased sensitivity and distortion of taste. Sensation of metallic taste is the most common, which is in around 20%. Patients also had a greater sensitivity for certain smells, including food cooking. Patients have reported a wide range for food preferences and aversions, often for the very complex tastes and sweet tastes. Patients with taste changes at the beginning were those who had less energy intake and greater weight loss. The nutritional assessment is the first tool for the correct setting of the dietological treatment for the neoplastic patients. Maintaining an adequate caloric intake with food diet, even if added by supplements, remains the most important challenge for improving the patient's quality of life. The switching to the use of artificial nutrition, both enteral and parenteral, is necessary when the patient is not able to eat adequately and if receiving anti-cancer treatment, and in those with an expected survival of more than a few months. In patients with an expected survival of less than a few months, a decrease in the invasiveness of nutritional intervention is recommended, with dietary counseling and oral supplements preferred. In patients with an expected survival of less than a few weeks, comfort direct care is the recommended approach, including alleviating first eating-related distress and other symptoms. The same review underlined a particular attention for use of omega-3 in cancer cachexia for their anti-inflammatory properties. Most trials suggested benefits of omega-3 on weight, lean body mass, and some aspect of quality of life, when given to patients receiving radiotherapy, chemotherapy, or chemoradiotherapy together. However, when given to patients not receiving anti-cancer therapy, no benefit was detected. ESPEN Guidelines 2021 strongly recommend use of oral supplementation for cancer patients malnourished. Oral integration is a non-invasive strategy that can improve quality of life and in the short term can affect answer in secondary outcomes. The value of this approach is not just to restore the reduced caloric intake, but also to provide food with therapeutic effects, such as oligoelements, immunomodulators, vitamins, and obviously omega-3. Between the strategies suggested to increase the lean mass are taken into consideration omega-3. However, other protective and potentially benefit effects are not considered for other cancer-related complications that could be potentially managed by supplementation with omega-3. Omega-3 must also be considered as an immunonutrient and are commonly used in the nutritional therapy of many pathologies, in addition to the plastic because of their wide biological effects. This figure shows the network of terms of recurrence based on publication data recovered from PubMed and highlights the numerous and important correlation between PUFA and many topical neoplastic disease. This systematic review suggests the utility of enriched omega-3 ONS to preserve muscle mass during chemotherapy and improve quality of life with stronger evidence to be found. Omega-3 can act as agonist of receptor coupled to protein Z, interfering with the paraneoplastic syndrome such as anorexia, cachexia syndrome, pain, and depression. Further investigation are still needed to assess the concrete benefits of integration on PUFA in cancer-associated syndromes. This other review also shows a selection of 10 out of 157 randomized clinical trials with patients in chemo or radiotherapy or combined therapy, treated with an integration with comparable dosages of omega-3. The results agree with the preservation of body composition, of the weight, and the improvement of the quality of life. This study compared a hyperproteic, hypercaloric supplement enriched with omega-3, fatty acids, vitamin D, and antioxidants with an isocaloric control supplement to be taken for us complementary with the diet in a malnourished patient with advanced pancreatic cancer. A total of 200 patients were randomized. The results showed that only the supplement with omega-3 could increase weight, lean mass, and better quality of life. This supplement had a good compliance for a particular fruit and slightly acid preparation. No tolerance for sweet taste in many patients with non-tolerance for sweet taste in many patients with dysphagia. Conclusion, however, indicated the need of further studies to examine the potential role of supplements enriched with omega-3 in the treatment of cancer cachexia. Same supplement assessed on 55 patients in double-blind neoplastic patients with non-small lung cells cancer, finding less adverse events and improved quality of life in the patients under treatment. At 1 year, in the group of patients with the nutritional treatment with omega-3, there were no deaths against in the control group. This review takes into consideration another aspect, the alterations of the microbiota in neoplastic patients and the correlation with systemic inflammation. This image illustrates the interaction of systemic inflammation, probably due to the progression of the disease, which leads to dysbiosis and dysfunction of the intestinal barrier. The dysfunction of the intestinal barrier leads to the increase in the permeability of pathological bacteria and endotoxemia that increases systemic inflammation and muscle waste. The studies show how chemotherapy or radiotherapy are associated with presence of microbial dysbiosis that leads to dysfunction in the intestinal barrier. Firstly, the intestinal barrier can be compromised by the production of bacterial lipopolysaccharides, leading to damage of the intestinal cell membrane and increased permeability. Secondly, the reduced percentage of bacteria producing butyrate thins the mucous layer. Third, intestinal concentration of immunoglobulin A are decreased after chemotherapy or radiotherapy. In the figure, we see how omega-3 attenuates the dysbiosis, chemo, and related radiotherapy. Taking omega-3 restores the beneficial microbiota, bringing as a result a consolidated mucous layer, restoring intestinal permeability and increasing the concentration of immunoglobulin, reducing the intestinal inflammation. So administering omega-3 should be an option in these patients. This old Cochrane review of 2007 underlined that supplementation with omega-3 may not be suggested in those patients with dysgeusia for problems of compliance due to the low palatability. Now, some oral nutritional supplementation contains omega-3, but they are usually have a sweet flavor, which for the taste distortion in chemo and radiotherapy is not usually well accepted. We have tested in the last period this particular support enriched with 2 grams of fish oil in a concentrated fruit juice with an acid and fresh taste. We are testing patients in chemo or radiotherapy or chemoradiotherapy already in treatment with other oral supports, but no more tolerant for taste distortion. These patients had different malnutrition state that we have tested with MNI test. After a preliminary easy palatability test concerning color, smell, taste, consistencies, we have started this particular formulation with 2 or 3 bricks a day to meet the energy need. In this first period of 6 weeks, we are observing a good tolerance to the supplement and a conservation of body mass index. So malnutrition is still a partially unresolved problem in oncological disease. Nutritional therapy must be taken into consideration early in all neoplastic patients in chemo and radiotherapy. If food intake doesn't meet energy need, especially due to gastrointestinal problems and alteration of taste, we must use artificial nutrition, starting from oral nutritional supports up to enteral and parenteral nutrition. Palatability of nutritional oral support is very important to be tolerated by patients in the long period. The first target is to stop loss of weight and especially the loss of lean mass, which is associated with a worse evolution of the disease and an impossibility to follow the phases of oncological therapy in the set times. The beneficial effect of cancer-related complication of omega-3 fatty acids, despite the physiopathological premises and hopeful in vitro and animal studies, need further studies. Especially, we need randomized clinical studies due to the lack of clinical evidence in literature. There is a lack of scientific evidence to establish whether omega-3 can significantly prevent or improve cancer-related complications. We must understand overall in which stage of oncological disease could be useful. And now I'll be available for any other question. Thank you very much.
Thank you so much for your wonderful presentation, Dr. Formelli, and sure there are questions, so let's directly jump into the question and answer session. And we have received a couple of questions via email here. And it says, according to your lecture, Dr. Fumetti, you have observed an improvement in the patient's compliance with the fruity oral nutritional supplement high in omega-3. And this is compared to what? And do you see clinical improvement as well?
So the period of observation is still very short, and you have seen that I've observed just a few people, a few patients. But the tolerance is good because it's really a change of taste, and so everybody knows that after a certain time, patients don't tolerate anymore the same taste and the same consistency of food and oral supplementation. So this is really a bit different. So we always change the single supplementation, and this is really different. It's not just a question of taste, but of acid against the sweet.
Okay, so there's a superior taste in the fruity taste versus the sweet conventional products we have. Okay, great. Thank you for this clear statement. And is the dose of fish of omega-3 in Remune fulfilling the required dose for stopping cachexia? This is our next question.
Okay. Essentially, there are 2 grams of omega-3 in each supplement. So it's not really possible to say now that the concentration of this omega-3 in the supplement is enough to stop cachexia in general, but it's a dose which is in literature it's given like a good dose, and so it's comparable with the best that we can find now for our patients. So maybe there are different combination of oligoelements inside. We have a lot of vitamin D with the Remune and like pro-immune element, and but we don't have others. But the quantity of omega-3 is enough. And, good.
Okay, so in other words, there is no miracle product, but this is a very good one, state-of-the-art product where we can feed the patients the sufficient amount of omega-3 fatty acids with.
Yes, and we can better the quality of life of the patients in the hard moment.
Absolutely, this is a very, very important point you've mentioning here. Well, I have another question. Is Remune in general better than a high protein oral nutritional supplement? So here the comparison between Remune and a high protein containing oral nutrition supplement is asked for.
Okay, we can say if a thing is better than another because we all need something specific in a certain period of our therapy. Of patient in a certain moment could need more proteins, more calories, or omega-3. So we must choose the best moment. We can mix, we can associate different kinds of oral supplements. We usually do this. So we try to reach an amount of calories and a certain immune pharmaconutrition. So if we need just calories and proteins because there is really malnutrition, we give other kind of the supplements. But in this case, if we see that inflammation is the main problem of the patient, I think that this is something to try and to associate maybe to the others.
So it's not a question of if or so, but it's more the question of taking the comprehensive approach again with the patient.
Yes.
So let me check if we have other questions. Yes. In gastric cancer patients, does the acid of the fruit juice resist the gastric acid?
Yeah. Yes, it's just a question of taste, not of pH of the substances, so of the support, and so no problem. Really, it's a question of mouth and of taste, not.
Okay, good. No, that's also very clear then. Well, let me just check with our organizers. No, there is no further question as far as I can see. So thank you, Dr. Fumetti, for this really great session with you. I really enjoyed that. And we'll take another 15 minutes break. And we will be back at, let me just quickly check the watch, at it's 4:25 now in Germany, and we will be back at 4:50. So you actually have a larger break. So enjoy, relax, and see you later at half an hour. Bye-bye. everybody. Hello, everybody, and welcome back to our final session, which will be the last one for today. And it's called Proteins and its Clinical Relevance. And within this block, we have the pleasure to listen and to learn from our speaker, Dr. Lauren Hill, who's joining us from South Africa today. Lauren has 28 years of experience as a clinical dietitian, academic and researcher with a PhD in physiology in the area of clinical nutrition. She will share latest insights on the importance of protein supplement in clinical practice on the example of a rather new product range, differentiating from food for special medical purposes by being classified as food only, namely Vitalimid. Lauren, the stage is yours.
Thank you to b.braun and the Aesculap Academy for inviting me to take part in these NutriTalks. So I've been asked to speak about the importance of protein supplements in clinical practice with the specific example of Vitalimid. It is normally patients at nutritional risk that we consider protein supplementation. And just to give some perspective on what is regarded as nutritional risk, the ESPEN definition of this phrase or statement is malnutrition of sufficient degree to impair outcome. So this is quite an important insight to have, that nutritional risk implies that nutrition-related factors are already impacting on clinical care and medical treatment and potentially on clinical outcomes of patients. From a clinical viewpoint, if you are a practitioner in the field, a dietitian or other clinical nutritionist of some kind, it is therefore important that body composition or some measure of lean mass or muscle mass or a proxy for one of these measures is incorporated into nutritional assessment of the patient in the clinical setting. It was more than 160 years ago that Florence Nightingale noted these quotes from her nursing notes, and she commented that a small quantity of beef tea added to other articles of nutrition augments their power out of all proportion. She questioned the reason why jelly should be innutritious and beef tea nutritious to the sick. And she commented on the nutritive power of milk and preparations from milk, saying that it was very much undervalued. So without knowing why, she had already noted in her nursing practice the value of protein for acutely ill patients. And it is lean body mass that is really our goal in terms of clinical nutrition support. There are significant negative consequences associated with the loss of lean body mass. A loss of only 10% of lean body mass will already start impairing immunity in patients who are hospitalized or otherwise sick, increasing their risk of infections. At a 20% loss of lean body mass, impaired healing will occur, including muscle weakness, leading to consequences such as inability to wean off of mechanical ventilation, and impaired immunity continues. At 30% of lean body mass loss, new wounds start to develop, such as pressure sores. Patients are more vulnerable to pneumonia and have poor strength, leading to poor ability to recover and to cooperate with rehabilitation. At 40% of lean body mass, this is usually incompatible with life, and mortality is almost 100% in such patients, usually from infectious complications such as pneumonia. So it is not only clinical consequences that occur with loss of lean body mass, but also important health economic consequences. Patients such as this tend to stay in the hospital for longer periods of time or stay in the ICU for longer periods of time, and therefore cost more to treat, and also do not tolerate their treatment as well as well-nourished patients, leading to more complex medical care. This graph here is an example of the very rapid and significant weight loss, particularly with cross-sectional muscle area, that we see in the critically ill patients. And in a short period of only 3 days, patients with multiple organ failure may have already lost 10% cross-sectional muscle area. At a week, patients with multi-organ failure may have lost between 10 and 20% of cross-sectional muscle area, and at 10 days between 20 and 30%. This is commonly seen if you work in the ICU. You will visibly see muscle wasting, especially in patients who do not receive adequate nutrition support. So survival outcome is related both to muscle area size, and this has been shown in many studies that patients who are able to retain normal muscle area have a significantly better survival outcome than those patients who have progressively lower muscle surface area or cross-sectional area. But it's not only muscle size, but also muscle quality or muscle density that is associated with survival, as you see on this graph here. Important to notice is that many other chronic diseases, especially those with an inflammatory component, also pose a risk to lean tissue wasting. And we think particularly of cancer, which is quite iconic in this way, but many other chronic diseases will also present with forms of cachexia or forms of lean muscle tissue wasting. In these cases, the loss of muscle mass tends to be slower and more gradual, but places the patient at similar clinical risks. Then, when we think of sarcopenia, the group of patients that comes to mind here is the elderly, and this graph shows that after the peak level of muscle mass that's achieved in the early to mid-20s, there is a natural fall-off in muscle mass that occurs with aging. This dark line at the top here is the best case scenario with a certain amount of retention of muscle mass and strength, and the bright green line, the worst case scenario, where elderly patients may have had surgeries or been acutely ill, which has ended up with lower muscle mass. And it's not only muscle mass, but also performance, physical strength, the ability to recover from illness, and also to live independent lives. It's important to notice that the elderly also require a lot more protein than their younger counterparts in order to promote protein synthesis, to blunt the loss of muscle mass, and to maintain anabolic drive. So large groups of patients that we are used to seeing in the clinical setting are at risk, and in fact almost everyone is at risk. And important to note that it is not necessarily body size or BMI that places a person at risk. Patients in the hospital setting have particular risk factors which add to the risk for muscle mass loss, including that they are immobile, they may be comatose or sedated, they may be bedridden or on forced bed rest for whatever reason. Therefore, they have low physical activity and the low capacity to do physical exercise and therefore promote protein synthesis in skeletal muscle. And at the same time have low anabolic drive due to altered levels of anabolic hormones as a result of their illness or surgery or metabolic stress. And then frequently in the hospital setting, patients have reasons for excess protein losses, including wounds of various kinds, stoma, open abdomen, and then medical treatments such as dialysis, which also remove amino acids and proteins. So these patients are at particular risk. If we take this scenario as the normal state, a person with fully intact muscle mass, and a normal size of the lean tissue compartment, these patients are very easy to notice in our hospital setting. They are at high risk. They often come to healthcare already quite wasted, and they have significant metabolic vulnerabilities. They are at higher risk for poor clinical outcomes and therefore need quite proactive intervention. But patients who look like this are also at risk. Overweight and obese patients may be overlooked because they appear superficially well nourished. But obesity makes screening a lot more difficult in these patients, especially with quite blunt methods such as measuring just weight loss or BMI. Loss of muscle mass is difficult to detect in such patients, and overt low muscle mass or sarcopenia can be missed in patients who are overweight or obese. So on these diagrams, you can see patient figures with different BMIs, indicating that you can be obese with a low muscle mass, you can be obese with a normal or a high muscle mass, and similarly with BMIs across the spectrum, the body composition is more important than the overall body weight, which is also why I said it is important in clinical practice to do some measure of body composition so that you're able to track lean muscle mass deterioration or improvements. So protein supplementation becomes important in the hospital setting, but also in outpatients with these risk factors. Over the past 10 years or more, there's been a lot of focus on what the recommended protein intakes should be for patients in the hospital setting. And here you're seeing increasing protein intakes in grams per kilogram per day, according to recommendations, from a basal level of around about 1 gram per kilogram in otherwise healthy or stable patients, all the way up to more than 2 grams per kilogram per day in patients who are in active recovery or physical rehab settings, or who are recovering from an acute severe illness. Most commercially available enteral and parenteral products only deliver in this range for most patients of ordinary BMI parameters, or for very small patients. And as a result, you end up often with a protein shortfall that is difficult to deliver without delivering a lot of excess energy to such patients. So once you are treating patients with requirements in these very high ranges, you do need a solution for the protein element. So I'm going to tell you a little bit in practice how we would supplement free protein in our setting in South Africa, where I work and practice. The first method is that it is common to use just the amino acid or protein chamber of a multi-chamber parenteral nutrition solution. So the glucose and the fat chambers are not mixed in, allowing the amino acid portion to be infused directly because the infusion port is in this chamber. So this can be very helpful if you have venous access for a patient. However, it is quite an expensive method and it does waste the two unmixed chambers. So the other option is to use protein powders, such as the range of Vitalimed products that you are seeing here, which I will talk a little bit about in different applications. So for protein supplementation in enteral feeding or in patients who are on combined enteral and parenteral nutrition but still have a protein shortfall, we would use a protein powder mixed into water in the ward or ICU setting, and then drawn up with a syringe and injected either directly into the feeding port of the nasogastric tube or PEG tube, if that's the case, or through the medication port. And here we would use a small volume bolus to deliver the protein shortfall in a single dose or in different doses during the day. What is clinically important here is that the product has excellent solubility in water and is very easy to mix by hand at the bedside, where you don't have a blender or an electronic mixer. And very importantly, that protein can be delivered in relatively low volumes, especially in the ICU setting. The second option for enterally fed patients, which we don't commonly use in South Africa, but which is possible, is to mix the protein in water in a similar way and then put it into some kind of a container for enteral delivery, either via a pump or by gravity feeding from whatever container. So this is the reason why it's not commonly used in South Africa, because there is an additional expense. But if this is a routine method in your own setting, then it is very possible to deliver protein supplements via this method. And in terms of the Vitalimed range, this product has shown the features and the excellent mixability that will allow it to be delivered via these suggested gravity or pump methods. The priorities here again are that you want a product that is excellent in solubility in water and that produces an appropriate viscosity that you can pass it through a feeding tube and is capable of gravity or pump flow. Secondly, and very commonly, is the use of protein supplements as part of oral supplementation programs. And here it can be included alongside other forms of oral supplements where other nutrients are also desired, or it can be given on its own. And since this range is flavored, it is suitable for use on its own as an oral supplement. In this case, our experience from clinical practice is that patients tend to prefer a product that has a milder taste, and particularly products that are less sweet or even that have a slightly savory note. And here again, smaller volumes are quite important because they might be having other supplementation of other nutrients as part of the diet. Taste is quite important in terms of acceptability to patients, and compliance with oral supplements is linked to the taste experience. So it's also very beneficial to have a range of flavor options. The second way that oral supplementation of protein is useful is if you can incorporate a protein supplement into ordinary food. So you're therefore supplementing the protein in various kinds of foods that patients are ordinarily eating. And the features of Eva Plus here of having a neutral taste, the fact that it can be heated and mixed with a variety of food, as well as tolerating quite a wide pH range, makes it a very practical advantage for patients requiring protein supplementation. Now, in the South African setting, we tend to do enteral feeding using this closed system, where we use almost exclusively in clinical practice commercially available, sterile, ready-to-use products that are delivered enterally through a closed system. However, there are low resource settings in the rest of Africa and in other parts of the world that might still be using some of these open systems. And particularly for hospitals or clinic settings where food blends or recipe-based enteral feeds are being used, a protein powder that delivers a high-quality protein actually is quite an important increase in quality delivery of nutrition support. It is actually very difficult to make a food-based blend which is high in protein and still have a good consistency to pass through an enteral tube. So a high-quality protein powder is very useful. It fills an important clinical gap if you are working in such a setting. And just by replacing food-based protein with a protein powder, you can significantly improve the range of recipes that are available to your patients and an improved consistency for delivery down tube feeding. Lastly, I want to just mention the vegan advocacy. There are increasing numbers of patients in the South African setting who are following vegan or plant-based lifestyles, or who are seeking enteral products or oral supplements that fit in with their philosophy of following vegan lifestyles. And I'm sure this is actually a small population compared to some other countries in the world which have much bigger veganism movements. But the ability to deliver a vegan protein powder that still is high quality is also an important gap and is going to be extremely useful once this Eva Vegan is available in the market for use not only as part of food supplementation, but also as a protein supplement in the hospital setting. So that is a new practical advantage as well, to have a pea protein powder available. Thank you. That's all from me. I will see you again in the Q&A session for further discussion or to answer any questions you might have. Thank you very much.
Well, thank you, Dr. Hill, for this wonderful presentation, and welcome to our final Q&A session of today. Actually, we have received quite a number of questions, and so let's get started, I'd say. The first one is, What is your recommendation for protein supplementation in chronic kidney disease?
I'm not sure if the person is asking about what type of protein or the dosage of protein. And obviously, it also depends on the stage of kidney disease, whether the patient is being dialyzed or not. The KDOCI guidelines, for example, give quite nice specifications for both the protein dose and protein types for kidney disease patients of different stages. In patients who are dialyzed, very often you do need to supplement protein. The requirements are higher than many people realize, especially if there are restrictions on the diet. Often if you're trying to restrict phosphate or calcium, you might also be restricting high quality proteins. So you might need to supplement protein depending on what kind of kidney patient it is.
All right, I think this is quite clear. So really, again, we have to look at the patients individually. There's the next question. With regards to inflammatory diseases causing lean muscle mass decrease, can you state which of the shown diseases like cancer, COPD, HIV, kidney disease are the most drastic ones in losing muscle mass?
Again, I think it depends on the stage of disease, how the patient has progressed, and whether they are being treated for the disease and how well they're being managed for the disease. We tend to think of cancer as being sort of the iconic disease for muscle wasting, especially gastrointestinal cancers, head and neck cancers. But what's very familiar in our setting in Africa as well is HIV and AIDS, especially if patients don't have access to antiretroviral drugs. So again, it sort of depends on the patient. Patients with COPD, for example, can get extremely wasted due to the increased work of breathing. So it varies, I would say. All of them are at risk.
Okay, understood. I think the next question goes a little bit into the direction I placed the first question at how much protein would you recommend for chronic kidney disease patients ranging on a stage from 1 to 5? And can you make a general statement on that, or does protein supplementation need to be looked at individually per patient? I mean, I'll leave it up to you to answer that again.
I think I've addressed it. The doses for patients who are not dialyzed or who are in stage 3 and pre-dialysis might be as low as 0.6 to 0.8 grams per kilogram per day. There are also differences. There's a distinction between patients who are diabetic, who can get a little bit more protein, and patients who are not diabetic, where your protein restriction should be a little bit stricter, even below 0.6 grams per kilogram per day. For patients who are in stage 4, stage 5 and being dialyzed, you can go up to 1.2 grams per kilogram per day, and even more if they get acutely ill while they are in that stage of kidney disease.
Okay, good. So once again, we have to look at the patient again. And the next question again is dedicated towards the patient. If my patient requires extra protein, do I need to take Vitalimit, or can I also use regular protein powders, which are available at supermarkets, discounters, or drugstores? And what is the major difference?
Well, I think that it depends on the region of the world where you are working and what the regulatory structure is. Because things that are available over the counter may not be regulated if they are offered as a food supplement, for example, and therefore you might be at risk of getting a product that is not the quality that it claims to be, or that is contaminated with items in there that you don't necessarily want. Whereas a product that is supported by an industry base, has at least internal regulatory quality standards. So that would be my comment about over-the-counter options. If you look at the range of protein powders that are available from industry, from b.braun and also competitor products, the exact amino acid profile is very well described. The quality of the manufacturing process and the sterility and so on is also very high quality. So I think it's more a risk that you take if you use an unregulated over-the-counter product, even though you might think you're getting the same amount of protein. That would be my comment about that.
Very well. And I think it's clear because sometimes it's worth investing into quality and spending money on that, especially when we talk about such kind of products. Well, when it comes to proteins, you know that there are a lot of people afraid of overdosing proteins in terms of harming the body, especially the kidneys, but particularly others having from the diseases which are related to this organ. So what do you recommend those people asking that question?
Well, I think that for many of the diseases that we are treating as clinical professionals in the hospital setting or in the outpatient setting, there are well-recognized international guidelines that guide protein doses. And as long as you're incorporating all of the sources of protein in the diet, so that would include the ordinary food intake as well as any other special medical nutrition products that you're using, and protein supplementation if you're including a protein supplement such as this range, as long as those fit within the recommended dosing guidelines, that is what should guide your clinical practice.
For sure. So it's worth, again, that's what we heard throughout the day, it's worth looking at the guidelines and sticking to them. All right, very well. How long do you actually recommend protein-rich diets or supplements after post-hospitalization? What would you say?
That is a very interesting question. And I think there is a little bit of a misperception that once the patient has left the hospital or once the patient is in a post-discharge recovery phase, the requirements for protein really are not that high. And actually what we're seeing now with sort of phased protocols for all of the nutrients and nutrition support in general is that patients who are in that post-acute and recovery phase often need more protein than they needed when they were in the hospital setting, especially if they're in a structured rehabilitation program, if they're having to do a lot of physiotherapy, physical rehabilitation. That is the stage also when they are entering a more anabolic metabolic state, and they are then able to utilize protein in a much more efficient way. So don't scrimp on your protein in those post-hospitalization recovery patients. And then you can continue. The question was actually about the duration. You can continue until they are either able to eat their requirements in protein with normal dietary intake, or until their rehabilitation program is no longer as intensive. So it could be weeks. It could be weeks.
All right. So it's actually even more important to have a look at those patients after they are discharged from the hospitals, because they are well watched probably, and afterwards you have to have an eye on them. So this is that's what I take out of your statement here. All right. We have one more question. How do you adequately monitor, respectively screen the protein status of a patient?
That is actually quite difficult. There are a number of methods that you could use, and depends again on the level of intensity of your setting. One of the so if you heard Dr. Ioni's presentation earlier, she also talked about ways that you can investigate sarcopenia, and she gave different functional tests that you can use, including hand grip strength. You could try to look at biochemistry. It's quite difficult to interpret. And then if you are working in a very sophisticated or maybe academic or tertiary setting, methods such as CT scans or the use of DEXA scans or bioelectrical impedance can help you to determine body composition and protein mass.
All right, very well. Then thank you so much. I think we don't have any more questions coming in. Let me just quickly check, but it seems like that we have answered all of them so far. But that doesn't mean that you cannot raise more questions in the chat and afterwards, also by email, if you want to address them to Lauren or the other speakers. It's really no problem. We will try to answer them at any time after the event. Well, thank you, Lauren. I just wanted to thank you for this wonderful presentation and the time with you in the Q&A session. It's been great pleasure to seeing you again. And also to the audience, it was a great pleasure having you with us, and we look forward to welcoming you and all of the other speakers soon. Thank you, Lauren.
Thank you very much. Thanks for having me.
Now, for all of you guys having the agenda in front of you, will realize that we are actually, in fact, moving closer towards the end of the day, so and the final closing of our NutriTalks event. But before we do so, let us a little bit recap on the different sessions we've heard today, the insights we got, and the key learnings of today's sessions by inviting our KOLs to shortly wrap up their key takeaway messages and insights for you to recall and benefit from.
So how it worked for us, relatively well. We've not had any issues with the comp system. It's quite easy to train. It's quite clear. It's quite easy to set up default regimens. It's quite clear to set up the ingredients that we're using within that on that site, and trying to add patients into the system was quite easy. It wouldn't let you copy over patients that already existed, so it would already tell you if they'd been discharged from that system or whether they were still already on there. We're working in the process of trying to get the prescribers to do the prescribing on the system. And one thing, obviously, I mentioned before was to do with the updates, and we have switched to quarterly updates, which works well for both. Or I think all of us, actually, including people working at b.braun, the IT services team, my team in technical services and quality assurance, to make sure that we are doing it on the correct time. It's not meant to interfere with patient care, and there like, you have to remember that there's so many teams involved in kind of installing and bringing it all together. Some teams that you don't actually think about, but they are obviously have a good number of input that they will provide in moving the process or the product forward.
The process of compounding of parenteral nutrition starts with the prescription of the regimen by the physician and ends with the application and documentation. This includes the order entry, human error-free transfer of data throughout the whole process, a compatibility check with EDCOM, a broad portfolio of disposables, including oxygen and light-protected multi-layer bags, an accurate preparation with Apex, and delivery to the patient. With Apex, b.braun offers a state-of-the-art compounder for a safer, more accurate and reliable preparation of parenteral nutrition in a comprehensive compounding package to support the pharmacist and facilitate the whole compounding process from prescription to administration.
Dear ladies and gentlemen, dear colleagues, it was really a great honor to be with you today in this NutriTalk event. My part was to show you the latest results and recommendations with focus on the lipid emulsions containing fish oil. As I mentioned, in critical ill patient we have the problem of inflammation. And what can we do to fight this inflammation? This can be done by using fish oil containing lipid emulsions, because we have seen that in different models, in marine models, using sterile peritonitis or ARDS, we can definitely reduce this inflammatory response. And also in all the clinical papers available today, we can see clear benefits by using fish oil, independent of EN or PN, but PN is very effective, of course, this way. We can see clear benefits by reducing the number of nosocomial infections, time-free of infections. We have good results in pancreatitis, in sepsis, and so on. And even in the latest meta-analysis of Prodelli, there are clear positive effects. We can do something very positive. We can reduce the infection rates. We can reduce the hospital stay of length, the ICU length of stay. And in addition, there are positive pharmacoeconomic results. Despite the costs of PN with fish oil might be a little bit higher, in the end of the day, by reducing the costs for diagnostic and treatment of complications and reducing the stays in the ICU and hospital, we can see clear positive results for the hospital budget. All together means we should use omega-3 containing lipid emulsions in critically ill patients. But if you want to do this, my recommendation is clearly start as early as you can. You should start with lipid emulsions as an integral part of PN and EN nutrition regimens. You should start with lipid emulsions definitely in the beginning of the ICU time, and please use the fish oil as early as you can. The recommendations are quite clear. In addition, 1.5 gram per kg body weight per day of lipids in total, including non-nutritional lipid sources, and the intake for omega-3 is between 0.1 and 0.2. You can achieve this dosage by using the product of Lipidem Lipo Plus as a bottle system or as a convenience bag system. These are my take-home messages for you. Again, I say thank you very much.
So, what take home from this lecture? We have seen that we have a phase-adapted nutrition with different nutritional requirements in the very early acute phases, in the late acute phase, in the chronic and the rehabilitation phase. In generally, energy and protein should be ramped up over 2 to 4 days to the targets. You should use indirect calorimetry for the determination of the energy expenditure, and only when this is not possible, then you can use the ventilate data from the ventilator, and if this is not possible, then you can use predictive equations, and we have seen it's around 20 to 25 kilocalories here. The hypercaloric feeding here is recommended when you use the predictive equations, so you can go just to the low side of this range, which I told you before. We also have seen and discussed for several times today that you should use omega-6 fatty acid reduced fat emulsions, but to really get the best clinical benefit, you should go to the third generation lipid emulsions like Lipo Plus and use N3 fatty acid enriched lipid emulsions. Of course, you can do this also with ready-to-use 3-chamber bags as convenient solutions, or with the compounding to really tailor the nutrition to the patient needs.
Malnutrition needs to be prevented because it costs the healthcare system globally millions. And home nutritional support is the way to do that, is the way to support patient recovery and to support quality of life. It can be simple and it needs to suit the patient needs, the patient lifestyle. The most commonly used method of nutrition support in the community is gastrostomy. And gastrostomy can be delivered simply with a syringe where you do bolus feeding, mimicking the way we eating. And in nutrition support, monitoring is extremely important, especially nutrition monitoring. We want to know the efficacy of our method. And muscle, the importance of muscle mass cannot be neglected. And in this virtual era, post-COVID pandemic, there has been so many ways that we can do this virtual nutrition monitoring, and these have been discussed in my talks. So a collaborative approach where we work with the patient is the most effective method to support nutrition in the community.
Patients receiving parenteral nutrition can be at risk of vitamin and micronutrient deficiencies if they are not timely and appropriately provided. And among vitamins, vitamin K has fundamental roles for coagulation and for bone health. So it should be always provided to patients undergoing parenteral nutrition support, not only in the long term, but also for surgical patients and patient in ICU setting. The recommended dose of vitamin K is 150 micrograms, and new multivitamin products can provide these amounts, which should be always supplemented. We have to remind that we cannot provide the right amounts simply with the 3-chamber bags. So it's important to take multivitamins and micronutrients into consideration to provide patients with the best qualitative nutritional support.
The presentation about the importance of nutrition for patients with ostomies and chronic wounds focus on the energy production process, and to have the most efficient energy production process, patients are required to eat 3 times a day at least some starches. Besides, for patients with ostomies, there is some food that are irritating foods, and the main 4 irritants are tea and herbal tea, raw vegetables and fruits and whole starches, milk and dairies, and sparkling water or sparkling beverages. So pay attention to the nutrition for the patients. They will be able to recover faster, and the healing process will be much efficient. Thank you.
Dear ladies and gentlemen, b.braun is committed to the treatment of malnutrition in patients with impaired kidney function since decades. And we offer products and services for patients for malnutrition since decades. Secondly, we are committed to a personalized, individualized, and customized treatment approach. And our compound bag is only one example of this commitment. So let's join our expertise. Let's share our expertise and go along and fight malnutrition in a joint activity. Thank you for your commitment.
Many patients with oncological disease have a poor nutritional status, which has a negative impact on clinical outcomes and life. From 10 to 20% of the deaths in cancer patients can be due to malnutrition. Weight loss increases the toxicity induced by radiochemotherapy and lower quality of life. Oral nutritional support is not just a way to give caloric intake, but also to provide foods with therapeutic and effects like omega-3. Secondary cancer complication could be modulated by omega-3, like pain, muscle waste, microbiota alteration, and depression. Nutritional assessment must be started early and optimized through artificial nutrition. Starting from ons, then should be pleasant to taste. We need further study to confirm the effect of omega-3 fatty acids on cancer-related complications.
So for me, the summary points on the importance of protein in clinical nutrition are that protecting and promoting lean mass and skeletal muscle is a central goal in clinical nutrition. Delivering the optimal dose of protein in nutrition support is an essential component of getting this right. Protein requirements are under intense debate, but suggestions and evidence are that many patient groups should receive more protein than what is currently achieved in clinical practice. So this under-delivery of protein together with lean tissue wasting is associated with clinical complications, worse clinical outcomes, and higher risk of mortality in a number of important patient subgroups, including the critically ill, acutely ill patients, surgical patients, cancer, the elderly, especially the elderly, frail, and those suffering from chronic inflammatory diseases. So protein powders are a very practical and clinically useful way to fill that protein gap and compensate for protein deficits that occur. And they can be used as supplements both to enteral and oral nutrition supplementation.
Thank you very much, everyone, for this great wrap-up. I hope that next to the key messages summarized by our speakers, you, as our participants, have also been able to add several other aspects of new findings and inspiring thoughts for your personal take-home learnings. And if one or the other thought needs a bit more reflection or recall, please feel free to make use of our platform for the next 30 days to rewatch all sessions presented at any time preferred. From my point of view, I can say that it has been an outstanding day, filled with diverse topics, nice speakers, food for thoughts to learn and reflect upon. It's been great to see that the topics being presented found appeal amongst you, as proven via the interactive, lively, and partially also challenging question and answer sessions. Hence, I would like to share a big thank and appreciation to both all our speakers included in this event and to all the participants for bringing this event to life. Likewise, I would like to say a big thank you to your organizational team behind the camera, continuously dedicated to make this event a success by ensuring technical smoothness and moderation support whenever needed. It's been a true pleasure to be your moderator today, and from a professional and also personal view. I hope you enjoyed this day as much as I did. Take good care and spare 2 more minutes also to listen to our heartwarming thank you messages from the b.braun country organizations around the globe. Goodbye and see you soon.
Hello, from Team Nutrition here in the UK.
I hope you've enjoyed NutriTalks.
We look forward to seeing you soon.
Hello from Argentina. Thank you for participating in our NutriTalks.
Hello from the Philippines. Thank you for joining NutriTalks.
Olá, pessoal do Brasil. Muito obrigada por se inscreverem aqui no NutriTalks.
Mabuhay! Pagbati mula dito sa Pilipinas. Taos-puso kaming nagpapasalamat sa inyong pagdalo sa ating NutriTalks.
Dzień dobry Polsko. Zapraszam do wysłuchania NutriTalks.
Hola a todos.
Gracias por conectarse a NutriTalks.
Los saluda Anayeli Silva
y Eric Carrasco.
¡Viva México!
अस्सलामुअलैकुम, मेरा नाम इमरान सईद है। मैं पाकिस्तान से NutriTalks में शमूलियत पर आपका शुक्रिया अदा करता हूँ।
Hallo vanuit 'n sonnige en warm Suid-Afrika. Ek hoop julle die NutriTalks baie geniet en ons sien julle binnekort weer.
大家好，我哋係 b.braun 香港，歡迎嚟到 NutriTalk。
Thanks for attending this NutriTalk week. Hope you enjoy it and stay healthy, stay united. Thank you.
Hello from Vietnam. I hope you have exciting time with NutriTalks.
Щирі вітання з України. Вітаємо. Дякуємо за запрошення до NutriTalks.
Hola, desde Colombia les queremos dar las gracias por habernos acompañado a los NutriTalks.
Muito boa tarde a todos os participantes deste evento da b.braun Internacional, os NutriTalks. O meu nome é Joana Varandas e sou Product Manager da Nutrição Clínica na b.braun Portugal e, por esse motivo, gostaria de deixar aqui o meu especial agradecimento a todos os profissionais de saúde portugueses que se disponibilizaram a estar aqui connosco com o mesmo objetivo de melhorar a saúde das pessoas através, particularmente, da terapia nutricional. A todos vocês, muito obrigada pela vossa participação.